See, okay. All right, so let's move on to Chapter 3. Chapter 3, the first part of Chapter 3, we're going to be talking about probably some stuff that you've had in classes before. We're going to talk about just some basic Mendelian genetics. We're going to do a Punnett square by show of hand. How many people have done a Punnett square before? Most of us all have in some sort of biology class. But then in the second half of the chapter, we're going to go on to some more complicated stuff that maybe you haven't gone into before. And then in Chapter 4, we're going to go and extend the Mendelian genetics and get away from sort of this simple model. The important thing to remember about the original Punnett square when we start doing these things is this is sort of the place that we embark from. It's sort of our ground zero. We are going to try to understand how certain patterns emerge. We're going to try to understand some interactions between alleles and things like that. But this is the simplest of the simple, right? The vast majority of the characters that are out there are not inherited in such a simple way. And that's probably why it took us so long to make any headway in understanding these patterns of inheritance, is because we were always looking at these more complicated things and it was misleading us. And so as I mentioned, I think at the first of the semester, we've been interested in genetics, or how things are passed on or inherited, for 10,000 years or so, but it wasn't until just over 100 years ago, really, that we made any headway in really kind of understanding those things. And it's a really remarkable how long it did take us, and that's one of the reasons. Okay, so let's start by looking at the chapter outline. Mendel, we're going to talk about Mendel's study of heredity. We're not going to go do too much into that. You probably had background in that in other places. We're going to do applications of Mendel's principles. We're going to go through, I found that even though you all have probably heard of the principle of independent assortment, the principle of segregation, particulate inheritance, those things, it's good to sort of clarify those things, because I know a lot of times students kind of know what it is, but it's still a little bit foggy in their mind. We're then going to do some genetic hypothesis testing. We're going to do a little bit of statistics. We're going to introduce you to our first statistical test, the chi-square test. And then in the end, we're going to jump, and this is kind of a typical pattern that we do the whole semester, and this is kind of a typical pattern that we do in genetics in general. We try to understand what's happening in other models, and then we try to use that and adapt it to try to understand what's happening in humans. And so then we're going to do a little bit on Mendelian principles in humans. So Gregor Mendel, he was a pre-scientist, 1882 to 1884. He published in the 1860s his paper on the principles of inheritance. It was not widely read, his publication, until the turn of the 20th century. It was sort of rediscovered. Credit to the people that DeVry and some of these other geneticists that rediscovered Mendel. They didn't sort of steal his work, which happens a fair amount in science, probably more than we're comfortable with. But they gave credit to Mendel, and Mendel is, sort of the quote-unquote father of genetics. His research was principally done on the garden pea, the common edible pea. He was able, during this time period, and really only until not all that far back, there were people that did science were either part of the aristocracy or they were part of the priest class. And that was because those are the only people that had time and leisure in order to think about all of these lofty principles, right? The common people were too busy just trying to eat, right? That they weren't able to do it. And so a lot of our scientific discoveries up until maybe 100 years ago, well, no, really the Industrial Revolution. So about this time, Gregor Mendel. Before Gregor Mendel's time, it was pretty much exclusively the aristocrats and the priests that were really doing science. And that was regardless of whether you're talking about the Islamic world, you're talking about China, India. It was, again, priests or, really small group of elites that were doing it. It wasn't until the Industrial Revolution and the creation of a middle class, really a strong, robust middle class, that you actually had science being done by, many more people and people that weren't necessarily born into a life of leisure or part of the priest class. And so Gregor Mendel was one of these classic priest/scientists. He, in his monastery, he had his garden plot that he could go and do these types of experiments. So let's look at exactly what his experiments were, and let's kind of point out some important characteristics of why he was successful, why eventually he was able to do something that had eluded mankind for a long time. Okay, but first let's look at a few genetic terms. We've already kind of talked about these terms. I just wanted to explicitly define these up here for you. Gene, most of us know what a gene is. I want you to begin to think about a gene on sort of that molecular level, this sequence of DNA, right, at a particular location on the chromosome. That location, the gene-specific location, is what we call a locus. Plural of locus is loci or loci. I say loci. Alternate versions, we've already defined this, just barely. Alternate versions of a gene is an allele. And then two new terms, maybe not so new. Homo and hetero. Homozygous means that the alleles at the gene are the same variety or the same version of the gene. Hetero means they're other, right? Homo means same, hetero means other. Okay, most of you are probably familiar with those terms. Okay, so here, this is just a diagram of the chromosome 1 from a tomato plant, illustrating these things. So the location, this is the locus. Just think of, okay, here's the chromosome. We don't have a centromere there, but just a certain area on the gene. These are probably bigger than the actual DNA sequence really would be or would represent on a chromosome. But here we tend, just so that everybody knows, later on when we see these diagrams, when they're different colors, we just tend to mean those are different alleles, right? This would then be hetero or homozygous down here at the bottom. Heterozygous. It's different, right? And the colors just represent those different alleles. Okay, so back to... Okay, the secret of Mendel's success was kind of in its simplicity. So before now, people had been trying to figure out, these complex traits, how these complex traits are passed down in humans or livestock or whatever. They were having a really hard time doing it. What Mendel did is he selected the right organism. Again, thinking about a model organism, what kind of characteristics do you want your organism to have? Short lifespan. What's that? They can't all be the same. You want to have variation, but you also would kind of like to have variation that you could kind of control, right? So you have a bunch of different populations that vary, but that you can control the interactions between those populations. Easy to cultivate, right? You don't want something that you're going to spend all the time just trying to keep it alive. And you want to design your experiment in a way that really isolates out single variables. That was a big problem before then, as people were trying to figure out the inheritance of not only of complex characters, but multiple characters at the same time. And so what Mendel did is he came in and he said, all right, let's make it simple. We have these garden peas that have been being cultivated for a long time. These garden peas have a bunch of different varieties because there had been, commercial food, production type interest in these garden peas, and they had a lot of varieties that were true breeding. And so you had these really short pea plants that they call dwarf pea plants that you could go to your local nursery and say, okay, I want a bunch of dwarf pea plants, and they said, okay, here's this variety, and this variety is true breeding. When we say true breeding, this is a term we're going to use a bunch this semester. It basically means that if you have a population of true breeding dwarf plants and you come back in the next generation, they're all going to be true breeding dwarf. They're all going to be dwarf. And then the next generation, dwarf, 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 dwarf. What does that tell us on a molecular level? That they all have the same allele, that they're all homozygous. Okay, so if it's true breeding, they're all homozygous. That was a benefit. Small, easy to grow, we talked about that. And Also, in sort of elaborating on what, was it Pat? Did you say that or did Maya say? Variety. Oh, and being able to control the variation. The pea plant was really good at a good organism, a good system for that, because the sexual organs of the pea plant were enclosed in a flower. So that meant that, one, you could either self-fertilize the pea plant or you could cross-fertilize. You could specifically make the crosses that you want. And so just to explain that, here I have a picture of an intact pea flower. If you see here in this part, inside of here, it's just all these petals are all kind of tightly compacted here. If you peel the petals back, what you find is you have both male and female genitalia in here. The carpel down here is the ovary where you find the eggs. The stamen is this part, the anther is this part. On the anther is where you get the little dusty pollen. What is pollen? Essentially, it's gametes, right? It's sperm. So next time you have hay fever, remember that. Makes it kind of gross. So it's basically sperm, right? And so most of the time, so in these pea plants, you're hermaphroditic, right? You have basically both of the sexes, and so you can self-fertilize, right? And so you can have the pollen on this anther fertilize the ovaries and the eggs down here, or you can cross-fertilize them. Okay. Most of the time they're in tight like this. If you wanted to do an experiment, if you wanted to cross-fertilize two eggs or two pea plants, what would you do? You would just simply manipulate them, right? And so you would peel these back and you say, okay, I'm going to take pollen from, let's say, this purple flower pea, and then I'm going to, and actually how he did it is think of like little brushes, little watercolor brushes. You come in here, you'd scrape the pollen off of that. You then go to the other part of the monastery where you have a garden of white flowered peas, and what would you do? You would go and you would put, you would sort of brush up here and you would try to fertilize that plant. Okay, could you have some complications if you did it that way? What might some potential complications be? What do you want to stop from happening? Yeah, you want to stop the self-fertilization. And so what Mendel did is he, to stop the self-fertilization, he would say, okay, here's my pollen donor, right? Here's the anther. In this case, we're looking at a dwarf plant, right? So you come up here with the brush and you get a whole bunch of pollen from the anther, and then you come to the stigma and sort of drape all that pollen onto there. But what has happened here? He's cut off the male parts, right? So that you can't have any pollen coming from here and self-fertilizing the plant, okay? So it's a really, really nice, simple system, right? So in that way, he could really control the kind of crosses that he was doing. And this was important because there's a lot of noise, there's a lot of background that could be confusing if you don't control your experiment in that way. So it's simple, but it's really controlled. And that's what made it so, that's what made sort of his experiments come out the way that they did. Okay, so here, let's just look at one of the crosses. This is what we call a monohybrid cross. Mono means what? The root of mono is what? One. Okay, so one, what is the one that we're talking about here in monohybrid cross? Maybe one gene, but let's go even more general than that. At the time, Mendel didn't know what genes are really. They're just sort of these hereditary factors. What, Elizabeth? Yeah, Elizabeth says characteristics. So we're just looking at one character. We'll shorten it. We'll say we're looking at one character. Okay, so here we have purple flower pea plant and a white flower pea plant. We start with these true breeding parents. What do we know about these true breeding parents now? About their alleles? They're all the same, right? So if we said this allele is little y, it's homozygous, right? This one is big Y, right? If we didn't do this cross, if we just let the purple flowers mate with purple flowers, what would we get? 10 generations down the line, purple. 15 generations, purple, right? It would all be purple. Okay, so he would take purple flowers, white flowers, he would do a whole bunch of crosses. He would then look at what resulted in those crosses. This is so here's the parental generation, the P generation. This is the F1 generation. The F stands for filial 1 generation. The root of that word, does anybody know what the root of that word means? Does anybody speak Portuguese?
It's
you do?
Or
Yeah.
offspring.
Do you speak Portuguese?
I do.
All right, so tell everybody what fila da puta means. That's where you get it, right?
It's like
son.
It's like a son of a bitch.
Oh, I just kidding. You weren't supposed to say it. Yeah, so philo, it's philo, right? Phil in French. Anybody speak French? That same root. It's son or daughter, right? Okay, so son. So this is basically the son or daughter generation 1. Then what's the second one? You're the first. I always ask that. Everybody says they know Portuguese, but then they never say it. You're the first one. Brave enough to say it.
You gotta do what you gotta do.
That's good. This class goes all in. I like it. So filial 2 generation, this is the F2 generation. Okay, so what happens in this F1 generation? You get offspring, and all of the offspring are purple. Okay? Would that be surprising during that day? What was the general notion of genetics before Mendel? Blending, right? Blending genetics. And so you would think, okay, all of them should not be purple. They should probably be a little bit less purple. But then you might say, okay, well, I don't know. Maybe it's just, we're thinking too much into it. Maybe it is just kind of purple and white and purple. Maybe it doesn't make it lighter purple. Maybe it's still the same kind of purple, right? What else does this tell us? What do we know right now in the F1? What information? And this is what I want to get you to start to think about. All right, what information can we glean from those data, that data, right? What's it telling us? Okay, so the first part of what you said, that there's a dominance, right? A dominance recessive interaction between what? Different genes or different alleles, right? So different alleles at the single gene. So this is telling us now, first generation, this tells us, okay, the purple allele is dominant over the white allele. Okay, so then what did Mendel do with the F1 generation? What would you do if you were Mendel? How would you do it? You just go break into the wine or something and do nothing. You just sit there for 3 days. Why? 4 days. Why? What do you have to do? Nothing, right? They all self-fertilize. So this is the easy part of the experiment. You can take a vacation, relax, let them all self-fertilize, right? They all self-fertilize. Then in that next generation, what happens? All of a sudden, this is the surprising part right here. This is the really surprising part. All of a sudden, that what we then identified as the recessive characteristic, right? The recessive allele, all of a sudden emerges. So it disappears in this generation and emerges in this generation. That was the big deal, right? All of a sudden, what does that tell you about blending inheritance? Can you get a whole bunch of purple paint and mix it together and get white paint ever back out of it? No. And that was an important part, and it became even more important during the 1920s, during the modern synthesis of evolutionary biology, because it said that these hereditary factors are inherited in particulate form. It's not this liquid blending, but it's just these separate little particulates. Okay, and so based on that, Mendel came up with 2 sort of ideas. One, that alleles are segregating or separating and then coming back together independently. And 2, that when they are together, say in that F1 generation, they don't modify each other, that they get inherited, they separate out, they segregate out and come back together in a particulate fashion. So those were 2 big discoveries. This first one, that alleles are separating or segregating independently from one generation to the next, right? Or from each other in a single generation, became known as the principle of segregation. Okay, so what did you do after that? If you were Mendel, what would you do? You did an experiment. What would you do? Do it again, and you do it a whole bunch of different times, right? And that's exactly what he did. And he went through and he did it a whole bunch of times. Do I have the purple? Okay, here's the violet, white flowers, F2 generation. He got 1,000 of them. So he did it so many times that he was counting 1,000 different plants. I mean, that's a lot. Again, how many of you do gardens? How long would it take to whatever plant to get 1,000 of? Pretty tough, huh? So he did it, but then he said, okay, now we have a pretty good idea of what's going on. Let's see if the same thing happens with some other characteristics. So then he looked at some other characteristics. He looked at tall plants and dwarf plants, round seed, wrinkled seed, inflated pods, constricted pods, and he did it here with the round seeds. You're going to get more seeds from a plant, right? So he was able to do how many? Was that almost 8,000 seeds? That's a lot of seeds to be generated. Here, again, 900 individuals. And so he did it repeated over and over and over again. But here was the thing that, again, was sort of surprising. When he began to look at the ratios of those characters of the dominant phenotype, so we haven't really defined phenotype. The phenotype is physical manifestation, right? It's what you look like. The dominant phenotype versus the recessive phenotype, what did we see? There was a 3-to-1 ratio in each of those. So he looked at these in a simple sense independently, then he gathered these data, and then he says, okay, I'm going to try to generalize it. What is going on? Why is there a 3-to-1 ratio? And this is where sort of the genius of Mendel is sort of illustrated. Going from that to saying, okay, we kind of think about it doesn't seem that big a deal to us in retrospect, because we're like, oh yeah, Punnett square, et cetera, et cetera. But he didn't have any of that. He didn't even know what genes were. He didn't know that there was necessarily 2 copies or anything like that. From that, he came up with this idea. And I think that is a pretty good, pretty massive mental jump. If you compare that to, say, like Darwin, like if you look at what Darwin proposed, most researchers after Darwin, they would say there were Thomas Huxley, actually related to Aldous Huxley, but Thomas Huxley, after Darwin proposed, evolution via natural selection, he was just like, that's so stupid. Why didn't I think of that first? Just because it was really easy, and once it was there, you're just like, oh yeah, that's a really, that's so. It's just staring you, right? It's just so easy and staring you right in the face. With the genetics, it's still kind of like, I don't know if I would, like Darwin, I could have done what Darwin did. But Mendel, I don't know if I could have made that mental jump like Mendel. So it's pretty impressive. So he goes to, and he does that. What does he then do? He says, okay, let's devise some sort of system to keep it organized. And this is kind of the system that we do. So let's take it through what we know. Okay, so let's take the tall and the dwarf. We now know that there's 2 different chromosomes. On each of those chromosomes, there's a gene, right? And then there's 2 alleles. And so what happens? This is where we explain the 3-to-1 ratio. So we have a big D, little d, a big D, big D, little d. What kind of gametes can this tall plant generate? Tall alleles, big Ds, right? What kind of alleles can the dwarf plant generate? Small alleles, right? So here's a parental generation. That's why all of your individuals in the F1 generation, big D, big D is dominant over little d, so they're all going to be tall, right? 100% of them are tall. All of them have the exact same, and this is what we call the genotype, right? The combination of the alleles of that gene. Okay, so now when the F1s either self-fertilize or you cross different F1s, what kind of gametes can this individual produce? It can produce eggs or sperm, right? Some sperm have a big D, some sperm have a little d, some eggs have a big D, some have a little d, right? And in what proportion are they going to generate that? How many big D's compared to little d's is that individual going to produce in its gametes? What's that, Elizabeth?
About 50-50.
About 50-50. And where does that come from? Think back to chapter 2. In meiosis, you go through, and this is actually a good exercise to do. Go back through and trace that and trace, okay, how come in an individual that starts out big D, little d, you get 2, let's say it's sperm, 2 sperm at the end that are big D and 2 sperm at the end that are little d. Okay, so then what do you do when you cross these? This is where we get the Punnett square. right, when we do the Punnett square, we take the potential gametes, right, we line them up on the, up here on top, we line them up here on the side, and then we bring them over, big D to big D, big D to big D, big D, little d, big D, little d, little d, little d. Then you then look at these genotypes, right? Genotypes here, big D, little d, give you tall, 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 and dwarf. What do we know about the tall is they don't all have the same underlying allele combinations or genotypes, right? So after all of this, we can come up with a phenotype, right? Tall versus dwarf, a genotype, the allele combinations that give you those phenotypes, right? The genotypic ratio, or let's look at the phenotypic ratio first, 3 to 1, 3 tall to 1 short, and then the genotypic ratio, which sometimes is the same as the phenotypic ratio, sometimes it's not, it just depends on how those alleles are interacting, a 1-to-1 genotypic variation. Okay. And here, for next time, or go through these vocabulary words, and you need to be familiar with all of these. We kind of already know when we come back on Friday, Tuesday, Monday, Wednesday, when we come back on Thursday, yeah, when we come back on Thursday, we'll go through, and that'll be the first thing that we'll do. We'll just make sure everybody understands the difference between homozygous, heterozygous, genotype, phenotype, parental, allele, segregation, all of these. Okay, see you later today at lab. Okay. Okay, so now we're going to talk about a dihybrid cross. What happens in a dihybrid cross? Somewhere back here I heard it.
Multiple characteristics.
Yeah, multiple, and in multiple we mean di, which means what kind of multiple? Two, right? So there's two characters that we're looking at. We could go on to a trihybrid cross. What would happen in a trihybrid cross? Three characteristics, quad, four, etc., right? So we could get some really complicated crosses in the sense that they're complicated in that we're adding characteristics, so we're going to have to track all these different combinations. And what you're going to see is as we add characters later on in this presentation, or in this PowerPoint, as we add characters, it gets more and more and more complicated. Okay, more and more combinations. It's not a linear growth in combinations, it's more of an exponential growth in combinations. But let's see, this is what Mendel did. So he became, really, he gathered a whole bunch of data with the monohybrid crosses, and then he said, okay, we kind of understand what's happening in these more simple situations. Let's up the ante, and let's look at two characters at the same time. And so this is a dihybrid cross. And so here, again, this is the parental generation. True breeding, when we say true breeding, what do we mean? Sorry, the zipper always comes out. So, what is it? True breeding? Homozygous, meaning what phenotypes emerge generation from generation? Which new phenotypes emerge? None of them, right? It's the same, generation after generation after generation. Okay, so we have yellow round. All of the P's are yellow, and they're round in this parental lineage. In this other one, they're all green and wrinkled. Okay, so up until this point, let's just pretend we know nothing about the interactions of these alleles with each other in the same genes, nor do we know anything about the interaction of the alleles between the two different genes, or the interactions, we could just say, of the two different genes upon each other. So we do an F1 generation. They all come out 100% yellow and round. Okay, what does that tell us about those characters? What does it tell us about the yellow-green interaction?
Yellow's dominant.
Yellow's dominant. Good. We already know that from the first generation, right? What do we know about the wrinkled round?
Round is dominant.
Round is dominant. So we're assessing the dominant recessive interaction. So we do that to begin with. Now, with the F1s, we let them self, right? So they all self. Could we draw out the genotype here? Yeah, we will here in a second, but let's just look at it sort of qualitatively right now. And let's just look at one of the characters, okay? We've already done these monohybrid crosses, so let's just look at the yellow-green. So here we have yellow, and then here in the F2 generation, we get these results, 315 yellow round, over 100 green round, around 100 yellow wrinkled, and 32 green wrinkled. Okay, let's compare green with yellow. How many yellows up there compared to green? Can we come up with a ratio? What's that ratio?
2 to 2.
So here we have 3. Here we have 400 roughly, right? And then roughly 400 to about 132. Well, what does that end up being? A little bit less. So somebody want to do it on a calculator? You know how to figure, we'll be doing a lot of figuring out these ratios, right? And so before you, do it on a calculator the first few times, but then after a while you'll be able to say, okay, around 400 and around 140, 130. 130 times 3 is what? 390. 390 is close to 400. So this is a 3-to-1 ratio. Have we seen a 3-to-1 ratio before? Where do we see that? In our monohybrid cross. So if we sort of close one eye and only look at one of the characters, we see, okay, it's a 3-to-1 ratio. Round to wrinkled, again, round here, 108, 315, a little bit over 400. Over here, about 130. Again, what is that ratio? 3-to-1 ratio, right? And so when you're figuring out 3-to-1 ratios, if you're doing it on a calculator, you're going to have to convert that to percentages, right? So how would you do that? Maybe we should do that right now, just so that we can kind of take first steps. So how would you do that? Let's just do the round versus wrinkle. So we have 315 plus 108, right? That's 423 of the round. So we write that up. And then how many of the wrinkled? 130, 30 what? 33. Okay, wrinkled. Add those together, what do we get? What's our total sample size? 556. Okay, so here's your total. So now we're looking at ratios. And so if it were a 3-to-1 ratio, then the recessive one should be what percentage? In a 3-to-1 ratio. Here's 3-to-1 ratio. You can do the same thing. Total 4. So the 1 is 1/4, 3/4. Ever see that? Okay, so it should be about a 25% ratio. So okay, so divide the 133 divided by 556. Somebody with a calculator or phone do that. What is it? It should be close to 25%. What does it come out as? 24%. 24%. That's how, so it's not an exact game here. You have to kind of, and as we go through and we do the labs, we'll do the Drosophila lab, and then we'll do the corn lab. Especially the corn lab, you're going to have to kind of figure out some ratios, right? Okay, so 1/4 is 25%, 3/4 is 75%, right? So divide 423 into 556, and you should come up with something close to 75%. If this seems a little confusing now, it's because you haven't done it enough. As soon as you do it a whole bunch, in a few weeks from now, it'll be secondhand. Okay, so now though, so we have 3-to-1 ratio when we're looking at just one character. But now we got to look at combinations of those characters, right? And so rather than just having 2 phenotypes, meaning yellow-green or round-wrinkled, just 2, we have to look at 4 different combinations of phenotypes. We have to look at yellow-round, green-round, yellow-wrinkled, and green-wrinkled. We can also place categories on these. This is yellow-round is which phenotype? Dominant or recessive? Dominant here and then dominant here. So we can kind of now start to talk about this as the dominant phenotype. This is the what? Recessive dominant phenotype. This is the dominant recessive and the recessive phenotype. And now we're going to generate a new set of ratios. So now what Mendel found out is when he did the dihybrid crosses between all of those characters that he had already looked at the single monohybrid crosses, you get this 9:3:1 ratio. He repeatedly got this 9:3:1 ratio. And so that's nice, right? Why it is nice is because you can flip it around. Just like you could flip around that monohybrid cross. If you didn't know anything about the genetics of some trait, let's say you're going to do some experiments on your squash plants in your garden, and you had, let's just say, yellow versus white summer squash, or spaghetti squash, and you did the same, you had true breeding yellow, true breeding white, right, and you decided, okay, I want to try to figure out how the genetics are working here. You cross them, and we'll just say hypothetically, I'll try to get away from what the real one is. Let's just say, or maybe it's anyway. Let's just say yellow, because I think white is dominant. well, let's just say yellow was dominant, and all of a sudden you get all yellow squashes out of that. And then you breed all those yellow squashes, and you end up getting, what did I say was dominant? White? Yellow. Let's say you get 75% yellow, 25% white. All of a sudden you get this ratio that you're familiar with, this 3-to-1 ratio. So now we're working from the ratio. We're now going to assume what about that system? And this is where understanding these ratios are more than just the ratios. They tell you something biological about the system. The 3-to-1 ratio is going to tell you, okay, what's going on here? What would you say if you had to describe that in a little short essay on an exam? What's going on here? How many genes are influencing this trait based on that 3-to-1 ratio? How many genes? 1. How many alleles? 2. So now we know it's this really simple system. We get this ratio. That's good evidence that this simple system is happening. We're saying, okay, there's probably 1 gene. There's probably 2 alleles. How do these alleles interact with each other? Because in that F1, we have the disappearance of it. Then in the F2 generation, we have the reappearance of that white. It's a dominant recessive, so it's a really simple system. This is basically the simplest system we're going to deal with. 1 gene, 2 alleles, dominant recessive interaction between the alleles. Okay, so remember that. Whenever you see that 3-to-1 ratio, that's where you're going to go. Okay, this is evidence that that's kind of the genetic system. And so Mendel did it the other way. He generated the data, got the ratios, and said, okay, this is described what's going on with those ratios. Now we can use those ratios and say, okay, this character is being inherited in the simple Mendelian fashion. Okay. Now if we're looking at 2 characters, right, and we get this 9-3-3-1 ratio, it tells us all of the same stuff, right, about each of those individual genes, but now it's telling us something additional. What's the additional thing that it's telling us? So 2 alleles in each of the genes. What's it telling us now about the interaction of those 2 genes? It's telling us that they're independent. Okay, you're getting this 9-3-3-1 ratio. This is just an expansion of that 3-to-1 ratio. And so it's basically telling us now that those genes are what we call assorting independently. This is Mendel's next principle that he comes up with by doing. Okay, back here. I'm going to try to get fancy. Oh, no, I know what I'll do. I'm just going to go to the next slide. Okay, so here we go through, we distribute all of those gametes up here along this axis, the top horizontal axis. We distribute those gametes on the vertical axis. Now, instead of a 4-celled Punnett square, what do we have? A 16-celled Punnett square. And then what do we do in order to figure out all of these genotypes? We bring it down, just like we were doing with our other Punnett square. We bring down this big G, big W. We bring over this big G, big W. That gives us this genotype in the upper left-hand corner of big G, little w, or big W, big W. What phenotype is that? Yellow round. All right, do this one. Bring this one over. Little g, big W, big G, big W. We get the phenotype, or we get the genotype, big G, little g, big W, big W. What phenotype is that? Yellow round, right? Okay, so that one's not in there. That's you pass the test. Okay, so what is this telling us? This is telling us that each gene is behaving independently of each other. Okay, and so if we go through and we count up the different ratios, right? Let's just count up the easiest one, the recessive, which is green wrinkled. How many up here are green and wrinkled? Just 1 over here in the corner. 1 out of how many? 16. You could then come up with a percentage, right? And so in the ratios, 9, 3, 3, 1 adds up. If you add 9, 3, 3, and 1, you get 16. 1 and 16. Let's just do another one. Let's do the green round. How many green round are there up there? 3. Right here, right here, right here, right? 3 over 16 ratio would give us a certain percentage. That's what the percentage is down here. Okay, so now we say, okay, this is our prediction. We go through, we look at our experiment, we generate all of our F2s, and we come up with a percentage that is somewhere close to that ratio. We'll show you that on the next slide. This slide just shows you that expected phenotypic ratio, right? With the monohybrid cross, we also talked about a genotypic ratio, right? Here's a different genotypic ratio. So how many different genotypes are there? 1, 2, 3, 4, 5, 6, 7, 8, 9 different genotypes. And here are all the different ratios. So this would be 1 over what? 16, right? You're still going to have, you have still all, think about that Punnett square. It's still a 16-celled Punnett square. 2 over 16, 2 over 16, 4 over 16, etc., etc. Once we get up into dihybrid and trihybrid crosses, the genotypic ratios stop meaning as much, but we could still say, okay, in that F2 generation, what percentage of the offspring do you expect to have a genotype exactly the same as the parents? That could be a good question. How would you do it? You go through, you would count up what's the genotype of the parents? Big G, little g, big W, little w, good Carl. You come over here, 4 out of 16. What percentage? 25%. So that could be something. But we're usually, when we get into more complex ones, we're usually more interested in the phenotypic ratios. Okay, makes sense? All right, this is the background. These are the data that were generated for Mendel to formulate his principles of independent assortment. It's just basically telling you that alleles at different genes are coming together and separating out independently. We now know that this happens when genes are located on different chromosomes, or genes are located a pretty good distance from each other on the same chromosome. And we'll get into that in a later chapter with crossing over and constructing gene maps and things like that. Okay, any questions? Okay, here, this is the slide that I was referring to before. We can go through and we can count it and we can look at these proportions, right? And what we see on these proportions is that you have an observed number, right? When you total up all the observed number, that 556 over here, if this population was exactly in a 9:3:1 ratio, how many would you expect to be dominant out of 556? How would you calculate that? So you know you have 556 individuals, and you know that you have observed 315 yellow round, 108 green round, etc., etc. You total those up, that's your sample size, 556. How many of those should be dominant if this system is exactly in a 9:3:1 ratio? How would you calculate that? Yeah, good. Did we hear that? So you take 556 and you times it by 9/16, right? Somebody want to do that for me?
313.
Oh, without rounding, what would it be?
312.75.
Okay, so 312.75. Can you get a 0.75 of an organism? No. So what do we do with that? No. That's what they did. They rounded up, and that's actually not what we want to do. We'll see is it's important to keep the expected ratio, right, in a decimal point when we start to do our chi-square test. We start to statistically test it. The book misleads you here. You should keep it in those ratios because as you can imagine, if you round up from 312.75 to 313, that's not going to affect you. It's not going to affect your outcome very much, right? But that's just a really slight deviation. But if you were to round up 34.25 to 35, how much of an increase is that? It's a lot bigger, right? And so we want to make sure, write this down because students always get confused because of this figure in the book. When you were looking at your expected numbers, we're going to keep them at 2 decimal points. You don't want if it's, there's the law of diminishing returns, right? The more exponent points you go out. So let's just keep it with 2 decimal points. And so when you go through and do that expected number. But what do we see here? Just qualitatively, we're not going to go through and do our statistical test yet, but qualitatively, what do we see? It's pretty close, right? 313, 315, 108, 104, 101, 104, 32, 35. It's really pretty close, right? Close enough, right? Why isn't it exactly? One, you can't have fractions of individuals, but why else? Why isn't it exactly? So large, so Elizabeth said smaller sample sizes. What happens with smaller sample sizes? What do we have to deal with? Random deviations, right? Random deviations in smaller sample sizes are usually proportionally bigger, right? That's why we like large sample sizes, because we diminish those random deviations, which are the background noise. Okay, and we'll get into a whole bunch. We'll get into that more when we start talking about chi-square test stuff. Okay, so now let's see if we can actually figure out and do a problem ourselves. Okay, so here we're looking at summer squash. White fruit is dominant over yellow fruit. Is that what I said earlier or not? I think I said yellow, but it was fictitious, right? And disc-shaped fruit is dominant over sphere-shaped fruit. Here I've given you those alleles. I want you to look at a cross between a true breeding white disc-shaped plant and a yellow sphere-shaped plant. And I want you to give me the genotypic and phenotypic ratios for the F1 generation and the F2 generation. So usually what I do is I have you do that on a separate piece of paper, and then a lot of times I have you hand them in. And so I like to see where we're at. You don't get points for it or not, but it is one of those things where I'll like say, oh, okay, let's say you were really close to a grade at the end of the semester, and I'll just keep them, and I'll say, oh, okay, well, were you generating these things in class? And I'll say, oh yeah, look, you're really trying hard. So one, the most importantly, I want to be able to assess where everybody is with these problems, and two, sort of an incentive for participation. Okay, so let's take like a few minutes and see how quickly. Everybody just first do A, and then when you're done with A, just sort of raise your hand. And so I'll wait till a few people are done with A, and then we'll do it together, and then we'll do B. Okay, so for the F1s, what was the genotype? All of them were big Y, big S, little s. Phenotypically, all of them 100% were what? Y disc-shaped. Okay, so then for the F2, you're going to self those F1s, right? And so you get big Y, big S, little s, crossed with big Y, big S, little s. What do you have to do first? Before you cross, you got to produce gametes, right? And so all the gamete combinations are going to be what? Remember, distribute them through, like I was trying to show you before. Big Y, big S, big Y, big S, big Y, little s, little y, big S, little y, big S, big Y, little s. Okay, you have 4 different gametes. Is this beginning to kind of look familiar?
That last one, little y.
Oh yeah, sorry. Thanks for catching me. I won't be the first time I do something like that. So make sure you don't be shy about saying I wrote it up there. Because a lot of times I'll even say it and write it completely different. So I'm just trying to keep you on your toes. Okay, so we have these 4 different gametes, right? Each of them producing 4 different gametes. What do we do next? Make a Punnett square. We throw those 4 gametes along the horizontal axis, the vertical axis. You bring them all in. You figure out the genotypes. From those genotypes, you then figure out the phenotypes. And what are the ratios that you get? 9, 3, 3, 1. Okay, how many of you? Let's see, where is it? Oh yeah, so hey, color coded. Nice, Max. So most of you, yeah, Punnett square, Punnett square, Punnett square, half of a Punnett square, a little bit of a Punnett square, another Punnett square. So most of you, okay, you were able to kind of generate that fairly quickly. What we're going to do here, and I'll just give you a precursor for where we're going with this, is that a Punnett square sometimes takes a lot of time. right? You got to go through and generate it. Can we just look at this and figure out, let's say, a phenotypic ratio, the dominant-dominant phenotypic ratio? We're just going to do it really quick, okay? Phenotypic ratio of the dominant-dominant. So, of the big Y, little y crossing with the big Y, little y, what percentage of those offspring are going to be white? 9/16. 9/16.
Yeah, like
We're just looking at one right now.
3/4.
3/4, right? So 3/4 of them are going to be, okay, we'll say, what's the probability that we're going to get a white? We'll just draw it out sort of mathematically in this cross, big Y, little y, with big Y, little y. It equals, we can even just do a little, go back to doing the easy Punnett square, the 4-cell Punnett square. If we did that, right, big Y, little y, big Y, little y, we see that big Y, big Y, big Y, little y, big Y, little y, little y, little y. 1, 2, 3 out of how many cells? 4. That equals 3/4. Okay. How many, what's our probability or what proportion in the next generation are going to be disc shaped? I keep spelling disc differently. It's going to be 3/4 again, right? What do we notice about this? If we times 3/4 times 3/4, what do we get? 9/16. There's that ratio. Okay, so, well, don't worry too much about this right now. I'm just sort of getting you primed for what we're going to do in a little while, a little bit more complicated stuff. Okay. So, another one, all right? In summer squash, we're going to do the white fruit dominant, right, over the yellow fruit, disc, etc., etc. Same system. Let's see if you can see how quickly you can do this problem. If a squash plant that is heterozygous for disc shape and heterozygous for white fruit is crossed with a plant that is yellow spear shaped, what will the phenotypic ratios be for the F1 generation? So now we're giving you the parentals, but they're not necessarily true breeding. Okay? So you're just doing this cross. What's that? No, you're not going to hand this one in because we'll do it together a little bit. So then this will be the one that you can see the results. Keep the results. But don't ask me that anymore. I want to surprise you. If you hand it in or not. Oh, but just never rip it off. Oh, I see what you're saying. Yeah, I guess you can ask that. What's that, Maya?
Is the second plant true breeding or is it heterozygous?
You tell me. Any questions? Let's move on. Okay, so the key points, we won't go over the key points. This just summarizes this dominant recessive interaction, segregation, and independent assortment. Now we're going to go to the part, most of you probably by show of hands, how many of you have already done Punnett squares, 16-cell Punnett squares, etc., etc.? Most of you, right? Now we're going to get to stuff that maybe you haven't done already in previous classes. So maybe you have, maybe you haven't. I don't know, those of you in cell, maybe you have done this, but we're going to sort of expand that. And so what we want to do, and sort of reiterating what I mentioned earlier, is that the benefit of these ratios and these models, right, is so that we can predict outcomes. When we predict outcomes, they're based on a certain model, right, a certain system, being like with that monohybrid cross. The model would be the first thing that we did with the purple flowers versus white flowers. What's the model that we use to predict that 3-to-1 ratio outcome? What's the system? How many genes? One gene. How many alleles? 2 alleles. What's the interaction? Dominant recessive. That's our model, right, in a loose sense. This isn't a mathematical model. We can make it into a mathematical model, but this is our ground zero model, right? From that, we can predict what we think is going to happen in the next generation. If we predict that and it ends up being that, what does that tell us? Oh, okay, our system's in that model. But sometimes you get outcomes that don't follow that, and that's just as, or maybe even a little bit more important, because then it says what? we have a different system here. So now we have a ratio. We got to try to figure out what's going on with this in this system. So we're linking these mathematical processes to biological processes, and that's when math is important, right? For us, we're all sort of quote unquote biologists in here, right, of some type. That's what's important. That's why we went into biology. We didn't major in math because math's okay, but it's not our favorite thing. But we want to use math to figure out stuff about biology. Okay, so Mendel's principles can be used to predict outcomes. We make predictions from our models. Deviations from the predicted model is going to tell us something about the underlying system. That's the sort of take-home message that I want you to think about for the rest of this chapter 3. Okay, so we have a bunch of different methods for predicting outcomes. And this is where it gets a little bit mathy, right? One is the Punnett square method. We should be pretty comfortable with the Punnett square method, right? We just did it for a monohybrid cross. We just did it for a dihybrid cross. We probably should be able to do it. We have all of the tools to do it for a trihybrid cross. What's going to happen? It's just going to get a little bit bigger, right? But then we're also going to look at a couple of other methods that you can use. And honestly, some students will really like the fork line method, and they'll use the fork line method to try to answer everything. Some students will really like the probability method. They'll use the probability method to try to answer something. I have answered these. You could do all of this too with the Punnett square method. As we get into more complicated stuff, most students haven't stayed with the Punnett square, but you could. You just have to have really big pieces of paper, right, to bring into the testing center. But you'll see that either of these two sort of are equally beneficial, or it just depends, right? Okay, so let's talk about those two other methods, the fork line method and the probability. We already kind of had a little teaser into the probability method. Question?
Would we have our own option as to
Yeah. So that's what I was sort of getting at. There'll be problems. You'll have your own option, and not only that, but there have been times when I have thought, oh, this is really easy based. This is how I would do it, and students have surprised me by using a different method. I was like, oh yeah, that totally worked. And so, it's going to be, and that's part of the skill set that we're trying to teach you in here. Is that you're going to have a way that you think about those things once you leave here and you're exposed to other situations, right? Where, let's say, you're in charge of hosting Thanksgiving dinner and certain people want to sit by other people but not other people. You may just pull out the fork line method and figure out things like that. Or more importantly, for those of you who are taking those pre-professional tests like the MCAT, like the LSAT, etc., GRE, you'll be able to use these methods to answer those questions. And for some of the rest of you, you might even use these methods when you go down to Wendover and try to earn your tuition money. All right, so there's lots of skills you're learning here. Okay, so Punnett square method, we just did this, right? We could figure out the phenotypic ratios. What's this model based on? I'm just sort of reiterating what we already did. We're not going to go through and do that. We know we're pretty comfortable with the Punnett square method, hopefully. Okay, let's look at if we have our Punnett square method, right, of those two characters. Let's expand that to 3 characters. And this is where these other methods come in handy. Okay, let's say now that we're going to do a cross between, we're going to do a trihybrid cross. So here, dwarf is little d, tall is big D. Again, those same characteristics that we were looking at before, yellow-green, wrinkled, smooth. Let's say we had an F1 generation, right? We had true breeding, dominant phenotype in the parental, one of the parents, recessive phenotype. We breed those together. 100% of the F1s have this genotype. Big D, little d, big G, little g, big W, little w. We then self that generation. What are the phenotypic ratios of the offspring in that F2 generation? Could you do that by the Punnett square method? What would you have to do? You'd have to produce the gametes, right? When we went from a monohybrid to dihybrid, we went from a 4-celled Punnett square to a what? 16 cell, this is also another skill that maybe you'll use on these tests. If we go to a 3 character, how many celled Punnett square are we going to have to use? 30, how many? 16, is it doubling? It's actually timesing it by 4. So you went from 2, so you went from 1, so you went from 4 to 16, right? So 4 times 4 is 16, not times 2. 16 times 4 is 64. Okay, so you're going to have a bigger piece of paper. You know, maybe you want to get a legal size pad. If we do a quad, if we do 4 characters, you're going to go from 64 to what? 256 cells. Could you do it? Yeah. So maybe, you know, maybe you want to go buy some poster board to bring to the testing center with you. If you decided, I don't want to learn how to do this fork line method or the probability, and you're perfectly welcome to do that. I'll even sign the poster board that you can take into the testing center. But if you don't do that, if you're looking at 264 cells, right, is there any possibility you might mislabel one of those cells? Yeah, there's a big possibility, right? And so this method is not only quicker, these other 2 methods, but they're a little bit more accurate. Students make fewer mistakes. Okay, so let's just go back to this, to the 3 character cross. All right, what are the phenotypic ratios? What are the phenotypic combinations? So before, we can also look at a pattern here, right? How many different phenotypes are we going to get? We could distribute them through. We can have a tall, yellow, smooth, right? What else can we have? Tall, yellow, wrinkled, etc., etc. And so we could generate all of the potential phenotypes. Should we do that? What do you think? Could we do that? So basically, you can, this is what I like to do, is I like to say, okay, here's one, we'll just call this gene 1, gene 2, gene 3, right? How many different phenotypes are there in gene 1? 2, right? There's either tall or dwarf. How many different genotypes are there for gene 2? 2, yellow or green, right? How many for 3? 2. So how many combinations? If we want to know all the different combinations, what do we do to figure out all the different combinations? Mathematically, just multiply them. Okay, 2 times 2 times 2. So 2 times 2 is 4 times 2 is 8. There's 8 different phenotypes. Okay. In the dihybrid cross, how many were there? How many different phenotypes? Yeah, dominant, 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 recessive, recessive, dominant, dominant, recessive, recessive. 4 in the di. In the mono, how many were there? 2, right? And so in the mono, there were 2. We see a pattern, 2, 4, 8. What's it going to be in the next one? 16. So it's not growing in this linear fashion as we add more and more characters. Okay, so there's 8 different phenotypes. We could even not even worry about it. We could list them as just the dominant, dominant, dominant phenotype, dominant, dominant, recessive, and we could draw all of those out if we wanted. Okay. So in order to do that, what sort of ratios could we do? Like, let's just, instead of going through and taking all of them, right? There's a whole bunch of them we could do. Let's just take one, and we'll try to figure this out. This one out. Okay. Tall, yellow, wrinkled. So there's all these different combinations, right? There's 64 different cells. It seems that we've run into a fairly complicated problem. One thing that you want to do in this class when you run into complicated, complex problems, the best thing to do is try to see if that complicated problem isn't just a whole bunch of simple problems added on top of each other. And so in this case, what are we going to do? We're going to say, okay, here we want to know what the phenotypic ratio of tall, yellow, wrinkled, but rather than trying to figure all those out at once, let's take this big problem and chop it up into a bunch of small problems. Let's just look at tall. Okay? So in this case, tall is the genotype is coding for tall, right? Big D, little d. big D, little d. We have big D, little d, crossing with big D, little d. In that cross, we can do a Punnett square for it, but now, maybe by now, we can do it in our brains, right, just in memory, or in our heads. If we cross that, how many are going to be tall? 75%, or 3/4 of them are going to be tall. One-fourth of them are going to be dwarf. Okay, does everybody see that? If you don't see that, make that little Punnett square. You have the one little d, right? And we'll even draw it as little d, and we'll draw this phenotype. There's more than one phenotype there, right? What's important for it to be tall? What allele do you have to have? A big D. So what I usually do is I just do big D and then a dash, because it doesn't even matter what that other one is. It just has to be some allele. It can be a big D or it can be a little d. So 3/4. So here, tall. How many are going to be tall? Draw out the three different phenotypes. And we can even say tall under there, yellow, and wrinkled. How many are going to be tall in the final outcome? 3/4 of them, right? They're going to have different combinations of those other genes, but you know of the total, 3/4 of them are going to be tall. How many are going to be yellow? 44. Right? So, yellow. We can even draw, okay, tall is going to be big D, whatever. Yellow is going to be what? What's the genotype? Oh, sorry. Big G, whatever, right? It doesn't matter. How about wrinkled? Little w, little w. If we just look at these, if we break this big problem into three little problems, big G, little g, crossing with big G, little g, how many are going to be yellow out of that? 3/4 again, right? If big W, little w, big W, little w, how many are going to be little w, little w? 1/4. 1/4. If all of these things have to happen, so I'm saying if an individual is yellow or tall and yellow and wrinkled, how do we figure out the total probability of that? When we hear and, what do we think? From those of you who've had stats, multiply. So you multiply these all together. What do you get? 3 times 3 is 9 times 1 is whatever. I mean, it's 9. 4 times 4 times 4 is what? 64. There's your ratio right there. If you wanted to draw out your Punnett square, you could draw it out, you go and count it, and there would be your ratio. Okay, so this is where we're going with it. Okay, so here, this is just illustrating that. Do tall versus dwarf, do yellow versus green, do smooth versus wrinkled, right? Figuring out, okay, how many of them are tall, yellow, and green. We did that phenotype over here. That's sort of the probability method. We'll talk more about the probability method. But here, what if I said, what about this genotype? You could do it the same way, but if I was just asking you one, you might be able to do it through sort of how I did it here. But what if I was asking you, what is the ratio of each of those? We know this one is 9/16, right? But what would the dominant one be? And what would some of the others be? If I was to ask you all of them, this is the situation where the forked line method well. Okay, this will be the last thing that we do today. This is what I'm going to show you. Rather than just in your head doing these 3/4, 1/4, what you do here is you have the same cross, right? Big D, little d, big G, little g, big W, little w. You separated them into 3 different smaller problems, right? In this first one, big D, little d crossing with big D, little d. In the outcome, it's a 3-to-1 ratio, right? So 3/4, 1/4. All you do is you go here and you draw out both of those phenotypes. Tall, 3, dwarf, 1. Does everybody see where we did that? Because that's it. You got to see this before we go on. Anybody not see this? Don't feel bashful if you don't see that. It's the exact same thing we did over here, only we're drawing it out. Okay, there'll be 3 tall for every 1 dwarf. Now let's go to the second one. The second one, see color. There's going to be 3 yellow for every 1 green, 3 yellow for every 1 green. And so if we had tall, what we do is we do 3, 1, 3, 1, and then we draw lines to it here, a line to it here, a line to it here, a line to it here. The third one, then we go move on to the third one. segregating for seed texture, round versus wrinkled, 3, 1, 3, 1, 3, 1, 3, 1, all the way down. And we have to link each of these to one back here, right? You could switch around and do whatever orders you want, but you're basically just creating this. If you flip it upside like that, it kind of looks like a phylogenetic tree in a sense, right? You're creating this tree. And so once you have all of those spelled out, you can go through and you can say, okay, how many are going to be tall, green, wrinkled? You go through and you times 3 times 1 times 1 equals 3. You put 3 tall, green, wrinkled. Okay? How many are going to be tall, yellow, round? 3 times 3 times 3 gives you 27. Okay? How many are going to be dwarf, yellow, wrinkled? Go through and you times those out. Okay, what do all of these add up to? 64. There's your 64 cells of that Punnett square. Here is your dominant, dominant, dominant. Your new ratio, this is an expansion then of that 9331 ratio. So here we had the 9331 ratio. Now we're expanding that to 27, 9, 9, 9, 3, 3, 3, 1 ratio. How if you, this one plus this one plus this one, they all equal 64. How many different types of phenotypes do you have up here? Come back over here. Oh, we erased it over here. The 8 different kinds. So there's all of these patterns. What does this now tell you? You're looking at 3 characters. You get a 27,999,3331 ratio. What is that telling you? What kind of system are you looking at? What? Trihybrid. How are they interacting? How are the alleles at each of those genes interacting? First of all, how many are there at each of those genes? How many alleles at each gene? 2 alleles. How are those alleles interacting? Dominant, recessive. And are they segregating independently? Yes. How are the different genes interacting with each other? 3 genes, 2 alleles at each of those genes. Each of those alleles are dominant, recessive. And again, just like that dihybrid cross, that's telling us that they are independently assorted. So here's another ratio. It's just an expansion of that 9331 ratio. Again, it's telling us something about the biological system. That's pretty important here, but even more important is this methodology. Okay? This is the methodology coming up with combinations. So, the take home then is I want you to do the same thing without looking at the next slide for this cross. Heterozygous at each of the 3 genes with the homozygous recessive. I want you to give me the phenotypic ratios of each of those. Phenotypic and just the phenotypic.
Are we going to be on Trihybrid? Are we going to do 4 genes?
Yeah, we're going to do, well, after we're through with these methods, we're going to do stuff where there's 7 different genes you're going to have to figure out. So, yeah, but do that, but first, baby steps, right? First, let's do this one, then we'll come back on Thursday and finish it off. Question about the exam? Yeah, okay. So, somehow I knew that question was going to come. For the exam, I don't like to rush a lot of this stuff, and so if we don't get all the way through this chapter can take a long time. Well, wherever we stop on Tuesday, I'll find a good place to stop, and I'll probably stop in a way that I will work out more problems. Like maybe the last part of the lecture on Tuesday, we'll just be doing problems. So try to go through and do as many problems as you can. We're going to get for sure through these 3 different methods, and so start looking at the problems in the back of the book of those 3 different methods. If we don't get through, what are we on right now? What slide? What number? Okay, so let's look. For those of you who weren't able to finish it, let's just go through really quickly and show again one more time just exactly how this fork line method works. And so here you have kind of a complicated problem, right? 3 different genes. You're going to have to track the behavior of those 3 different genes through a generation, and rather than trying to do it all at once, you're going to take this complex problem and do what? Simplify it. You're going to take, rather than trying to address all 3 genes at one time, you're going to address each gene individually. And so here, the first one, big D, little d, this is a tall pea plant, right? You're going to cross it with the dwarf pea plant. Big D allele is dominant over little g allele. And so if you ignore the G's and the W's, look at just the big D, little d, little d, little d cross. You could do a little Punnett square, right? Does anybody need to do that? And it's okay if you do. Maybe we should do it. So we're sort of combining the different methods, right? Big D, little d, little d, little d. What's the ratio? I imagine some of you did this. Why? Because if you did the 2 to 2 to 2, you would see here, okay, 2 to 2 ratio rather than reducing it to the 1 to 1. So here you have this simple Punnett square. You have 2 big D, little d's for every 2 little d's, little d's. It's the same as a 1 to 1 ratio. You then come down here, 1 to 1 ratio. We're talking, Chris asked a question just a second ago, is it phenotype or genotype? We're talking what? Phenotypes, tall to dwarf. That's the ratio, right? You go to the next one. Round versus green. You could do another Punnett square, a little simple 4-cell Punnett square again. Do that ratio. What does the ratio come out to be? The same thing, 1 to 1. You then do it with seed texture, round to wrinkled, 1 to 1. We can all do this multiplication, right? 1 times 1 times 1 is what? 1, right? You do it for all of them. What are the percentages now? So now we have a 1 here at this last column. We have combined phenotypes, 1 to 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 1. If we had to come up with the expected percentage of, let's say, tall green round plants in the next generation, how would we do that? Say it a little louder.
1 out of 8.
Yeah, 1 out of 8. 1, 2, 3, 4, 5, 6, 7, 8. 1 divided by 8. Right? What is it? 12%? 125. Yeah. Yeah. So 12.5%. Okay. So each of those, that's our expected. This is what we call a test cross. Carl, question?
Do you want us to reduce every time?
You don't have to reduce, but if it's on an exam and it's a multiple choice, you should be able to reduce it yourself or work in between those two so that you can pick the right answer. If it's on a part of the exam that it's not multiple choice, then I wouldn't mark you wrong for it not being reduced. Or if I did accidentally, you could argue that point and you get it. So no, it's the same thing, right? You don't necessarily have to reduce. And I know that people are going to do it different methods. Some people are going to use on some of the problems a fork line method. Some are going to use probability. And so I have to be like lenient in how I score them because I'm sort of focusing them and I'm stressing, learn these methodologies and figure out how to do these things, this sort of skill set on your own. And so I don't want to just do it this one sort of prescribed way. Question?
There's no difference between test cross and intercross.
Yeah, so what is a test cross? Why do we call it a test cross? So this one we say, okay, this is a test cross. The intercross, we were intercrossing those F1s, right? A test cross is named a test cross because you can test a genotype with it. So let's just look at one of these. So let's just look at this first one again, dwarf versus tall. Let's say we don't know that genotype. Let's say you find, in somebody's garden, a tall pea plant, and you wonder, wow, this is a tall pea plant. You know that tall is dominant over dwarf, right? So you could go and try to figure out the genotype by sequencing it, or just say you don't have very much money. What would be an easier way to do it? How could you figure out if that tall was either big D, or big D, little d? You can't see those alleles, right? You can't see those alleles, but can you figure it out in another way? What's that? Yeah, so if you have a dwarf, do you know the allele composition? Do you know the genotype of a dwarf phenotype? Yeah, because it's recessive. You know that's little d, little d. So all you have to do is you have to cross the normal height plant with the dwarf plant. And if you did that and you got, let's say you reproduced 20 individuals in that next generation, and all 20 individuals were normal or tall plants, what would that tell you about the genotype? It's homozygous dominant. If you got a ratio of about 10 to 10, or maybe 9 to 11, tall to short or short to tall, what would that tell you about the genotype? That it's heterozygous. And in that way, you're testing it. So this is a test cross. This is a test cross at each of these 3 genes, because we got this 1 to 1 to 1 to 1 to 1 to 1 to 1 ratio. What does that tell us? That, say we didn't know it. Say up here we just knew we had the phenotype, but we didn't know that there was a little d there, a little g there, or a little w there. Because of these results, we would be able to say, yes, in fact, in this the parental genotype here is heterozygous. So that's why we call it a test cross. Okay, because we can do sort of classical genetics, and it will tell us what the genotype is of that individual. And you don't have to know anything. You don't have to go into a molecular lab. You don't have to know anything of that. Anything like that, you can just go into your garden and do it. So now let's move on to probability. Here we're going to have to get a little mathy on you, but not too much, right? And in order to do that, we're going to have to define a few terms first and sort of explain kind of a perspective, or explain how we're approaching this. And we're not going to get too much into it, but we're going to get a little bit into it. Okay, so when we're talking about probability, I want to define 2 different things. One thing is an event, and one thing is a sample space. So those of you who have had, probability and statistics, I think there's a class in the math department, you probably have gone through this before. You probably know this sort of nomenclature, but for those of you who haven't, we're going to define it here. So an event is any outcome of a process. Okay, so any sort of process, there's an outcome to that. We're going to call that an event. So what could be a process? Here, I brought 3 nickels, because we make big bucks here as professors at Weaver State. I have a nickel in my hand, right? I flip this nickel. Is this a process, flipping a coin? Yes, this is a process, right? I flip the coin. What happens? I slap it down on the desk, and I get a tails. What is a tails? That is the outcome of a process. Process is flipping the coin. The outcome is tails. Okay, the sample space is a collection of all of the possible events, right? So if I flip the coin, I get a tails. I can say, okay, is that an outcome? Yes. So I got a tail, right? Let's say I went over here and I flip it again. This time I get a tails. Is that an outcome? Yes. I could write it up there. Oh no, I already have a tails. Okay, I flip it again. I get a heads. Is that an outcome? Yes. Yes. So that I can either get a tails or a heads, right? Does everybody see that? So this is my sample space. This is all of the possible events, or all of the possible outcomes. Okay? So now, here, this would be the sample space. Each of these individually is the event. This is where it ties into probability. The probability of something is the probability of the event is the event over the sample space. So if I go over here, I know this is the sample space. I can either get a tails or a heads, right? And I go and I say, okay, I want to figure out what is the probability. And this is kind of, overly simplified, but we're going to start overly simplified. And I say, okay, what is the probability that I get a heads? How could I figure out that probability? Here we write probability as P, and then in parentheses, the probability of getting heads is equal to the probability of the event over the sample, the event over the sample space. What's the sample space? 2 H and tails, right? 2. I'll even just put it up here. H and tails. H over H and tails. 1 over 2. What does that mean? What is that? 1/2. Okay, so that's probably not too surprising, right? So we can flip coins. Those are an event. We can then draw it out, and that's how we're defining a probability. Okay, does that seem not too overly mathy? There's a few sort of terms let's continue on to another example though. Okay, so let's still stick with coins, and let's say, okay, I'm not just going to flip one coin, but I'm going to flip 2 coins. Okay, so if I were to flip 2 coins, right, I could get what? What are the possible outcomes of getting if I flip 2 coins?
All heads.
So you get heads, right? What else can we get? Tails. What else can we get?
Tails, heads.
Tails, heads, right? What else can we get?
Heads, tails.
Heads, tails. There's an order to it, okay? Heads, tails. So if we could then calculate the probability. Okay, what is the probability? What is this? What are we defining this? All of this as?
Sample space.
Here's your sample space, right? Each of these are individual events. Let's call this event 1, event 2, event 3, event 4. What is the probability of getting tails, tails when we flip both coins being tails when we flip 2 coins? 1/4. How would we write it out just so we understand what's going on? Tails, tails is here, right? Tails, tails over, and we'll just say sample space. That would be tails, tails, heads, tails, tails, heads, heads, tails. How many are in the sample space? 4, right? How many tails, tails? 1. Okay, 1 and 4. What is the probability of getting a heads and a tails, regardless of the order? 1/2. Okay, because why? We still have our same sample space. That sample space is equal to 4, right? But we can either get heads, tails, or tails, heads. That's 2. 2 over 4 is equal to 1/2. Okay, so we're doing this with coins. You can go through and you could, flip them 100 times and you can mark them down and you should get somewhere around 50 heads, tails outcomes. You should get around 25% or 20 tails, tails, and 20 heads, heads. Okay, what does that have to do with genetics? That 1/4, 1/2, 1/4 is something that we've seen in genetics, right? When we do our test cross of a single individual, right, or when we do it with a single character, and not a test cross, sorry, when we do the intercross of a big G, little g, we do our Punnett square, right? Big G, little g, big G, little g. What do we get? Those same ratios. 1/4, 1/4, 1/4 here, 1/4 here. Add those two up and get 1/2. So it's just like flipping coins. So selecting gametes, what is the process in this? It's a little funner than flipping coins. What is it? Reproduction. That's the process, right? Reproduction is the process. What's the outcome? The outcome is one of these 4 genotypes in the next generation. Okay, so these are all of the possible outcomes, right? All the possible events that result in the process of plant reproduction. Okay, now that one's not as exciting. Okay, so are we all good with that? Any questions there before we go on?
So if we did it this way, rather than what we just did before, we have problems.
No, you can do it either way you want, but we're not quite done with this way yet. We're gonna go. Yeah, but yeah, you can use whatever. And I actually prefer that you select sort of the way that seems most logical to you. And from experience in the past, some students will like one way and some students will like the other way. But it's just a skill, right? It's just a skill set for you to have. And what you'll find is you can also, go beyond just genetics. Here we're doing coin flipping, right? So coin flipping, and we're just likening it to gametes. It's the same sort of thing. And so if you have a coin flipping problem, you can use it, later on in that. And that's why I said these skills are for those professional tests. Okay, here's another outcome. Here's another way to look at it. So we're going to kind of expand it. This is another process, sorry, with outcomes. What is this process? Throwing darts, right? And let's just pretend, just so that the analogy or whatever is understandable. We have some sort of mechanical dart thrower in the back, and it will randomly just throw it at here, but we won't count darts that hit outside of the target, and the bullseye is still just split into each of these regions. Okay, so what is the process? Throwing the dart. What is the outcome or the event? Hitting a certain number. All right, does everybody see that? Can we calculate the probability of the event over the sample space? How do we do that? Mathematically, how could you do that? Could you tell me the probability of hitting a 4? Carl says 1 out of 20. Okay, how would you do that mathematically? And could you prove that? So there's total number of 20, right? And then you divide it by one of them, right? What would happen if, you know, you say you got a dartboard from Kmart, made in China or something, and the 4 space was a little bit bigger? Then, well, how would that change it?
It increases the probability.
Increase it by how much? How could you calculate that? What are we looking at? What would you have to calculate? Yeah, good. So you're going to calculate the area, right? So just the area of this little pie right here. These areas all happen to be the same, but you would calculate that area here, come up with a number, divide it by what? The total area. So that, this is sort of a more graphical way of thinking about probability. This area over the total area, and you'd be able to come up with a probability. Okay, so now this is an example where all of the areas happen to be the same, but we I forgot that screws up the recording. Oh well. So this is called the multiplicative rule. Probability of getting the king is 4 in 52. Probability of getting the club is 1 in 4. Probability of getting a king and a club, we multiply 4 over 52 times 1 over 4, that equals 1 over 52. This is the multiplicative rule. This is just like before in the slide when we saw the probability of getting area A. What were the overlapped areas? I can't go back now because I've messed up the recording. A and B is a probability of one times the probability of the other. Remember, that gives you that overlapped area. So it's just a mathematical way to calculate that overlapped area just based on the probability of each of those events, A and B, because intrinsic to that probability is that area, and so that's just a way without actually going through and doing geometry or whatever and calculating that area, or trigonometry, whichever math that is, calculating that area. You can just do it simply mathematically. Okay, so let's go. This is the multiplicative rule. Let's look at the other example. A or B. That the other one? This is the, oh no, A and B. Oh, if A and B are independent, what's the probability of getting A or B? So whenever in probability, whenever we see or, what do we think? And means we multiply, or means we add. Okay, so if events A and B are independent, the probability of getting at least one of them is denoted by the probability of A or B. If we add A plus B, we get the total area of those two. What have we done? We've counted twice that overlapped area, because that overlapped area was, you know, embedded in A and it was also embedded in E. So now we have to minus out that overlapped area. How do we calculate that overlapped area? We times A times B. Okay, so probability of A plus probability of B minus the probability of AB. Okay, so example, what's the probability of drawing an ace or a heart? So let's do that one. Probability of drawing an ace is what? 4 in 52, right? Well, I guess I don't have to rewrite it. Probability of drawing an ace, 4 in 52. Probability of drawing a heart, or a heart, is 3 in 52. What is embedded in both this one and this one? The ace of hearts, right? Ace of hearts is counted in this, 13 of the 52. Ace of hearts is counted in the 4 of 52. So to find the total probability of pulling an ace or an heart, we go through 4 out of 52 plus 13 out of 52 minus 4 over 52 times 13 over 52 or 1/2. That equals 16 out of 52. So you could go home, take a deck of cards, pull them out, pull out all of the aces, pull out all of the hearts, and you should have 16. Okay. So you wouldn't necessarily be able to calculate that just based on your knowledge without knowing just based on your knowledge of cards without knowing this additive rule. Okay, is everybody good with that? So okay, let's sort of move on to more we have to mention. So sometimes we see, if you remember when we said, what is the probability of A or B when they don't overlap, we just add those two. So we don't have to necessarily minus out the probability of both because there's no overlap. Or we could factor that in to be consistent, but the probability when there's no overlap between A and B, this is when they're mutually exclusive, when there's no overlap, then that probability is 0, so you're just minusing out 0. Okay, so if they're mutually exclusive, meaning there's no overlap, we do the probability of A or B, it's A plus B minus the probability of A and B, there's no overlap, so that's minus 0, so we just kind of forget about that. So this example would be, what is the probability of drawing an ace or a king? Probability of drawing an ace is what? 4 in 52. Probability of drawing a king is 4 in 52. What is the probability of drawing an ace and a king at the same time? 0, you can't. You're either going to draw, you can't pull out one card and it's an ace king, right? The probability is 0, so you minus out that 0. So add those two up. 4 in 52, 4 in 52 equals 8 over 52. Okay, makes sense? All right, so now let's so I just want to establish sort of that foundational sort of probability stuff. Now let's use that to predict some of these probabilities. Okay, or predict some of these crosses rather. Okay, so use probability theory to predict the probability of this self-fertilization, this intercross of a pea plant, big G, little g, big W, little w. It's going to self-fertilize, right? This is, let's say this is an F1. What's the genotype of that? Is it round or wrinkled?
Yellow.
Round. And is it green or yellow? Yellow. Okay, so we have a yellow round and a yellow round. What is the probability of getting a green, wrinkled progeny? Don't do a Punnett square. Don't do a forked line. Let's do this for probability. So what do? We first want to just draw out what are the events, okay? What are the events here? We have to first know the genotype of that one, right? And so green and wrinkled, do we know the genotype of that? Little g what?
Little g, little g.
Little g, little g. So in this next generation, we're going to have a little g, and then wrinkled is what? Little w, little w. Here are 4 events. Okay? What is the probability of holding a... We'll go slow right here, even though it might be too slow, but that's okay. I want to be really explicit so you understand what's going on. Okay, what is the probability of this individual donating to this genotype of little g? What is it? Just this one right here. You got a little ahead of us. This individual going to donate that little g. They can either give a big G or a little g. What's it going to be? 1/2. Okay. What's the probability of this individual, even though it's the same individual here, right? Donated another little g? 1/2. What's the probability of a little w there? 1/2. What's the probability of getting in that event a little w? 1/2. So what's the probability of this event? And if you want to calculate the probability, well, how do you calculate the probability of this event and this event and this event and this event? What do we do mathematically?
We'll multiply.
Multiply them all out. Everywhere I said and, you put a multiplication sign. 1/2 times 1/2 recessive genotype. That's that bottom right-hand corner. Here now we've eliminated having to draw out that Punnett square. Could you do that with the fork line method? Yeah, you could do it with the fork line method too. Maybe you want to, draw that out, and you could do it. Okay, but we'll go. That'll be a nice little homework thing to do. Go back and do it. Let's do the second part though. Homozygous yellow or homozygous brown.
Heterozygous.
Oh, heterozygous. Sorry. Homozygous yellow or heterozygous round. So we have the same mating, right, going on, and we need to get one event, right? What's this one event? Here, homozygous yellow. What's the genotype for homozygous yellow? Big G, right? Or, what's this other event? Heterozygous round, big W, little w. Okay. Whenever we see or, what do we do? Add, right. You guys are on it. Okay, so in this cross, we're crossing big G, little g. What's the probability? We're not going to do each of these individually this time. We're just going to do the whole genotype. What's the probability of getting a big G out of that cross? Did you ever see 1/4? Could we cheat and do a little Punnett square here? Sure. Big G, little g, go through, big G, little g, little g, little g. What's the probability in that cross of getting a big G? 1 out of 4. Okay, so 1/4. We'll even keep it a little consistent. I'm not going to say 1/4. I'm going to say 4 out of 16, just because it'll make more sense later. Okay. What's the probability of getting a big W, little w?
2 and 4.
1/2. What's that?
2 and 4.
2 and 4. Or how about 8 and 16? Want to do that? 8 and 16. Okay. So whenever we see or, we add. What's the probability of getting a big G, or a little w, big W, little w? Add those two up. That equals 12/16, right? If we go to the Punnett square, what would we see? 12. I can't do it now because it's recording. What are we forgetting? What's the additive rule? One event plus the other event minus what? The overlap. How do we get the overlap? Minus the probability of getting big G, big W, little w. Okay, so now can we calculate the probability of getting big G, big W, little w in that cross? What do you think? Should we do it separately over here? Everybody have that in the corner? Big G, and then heterozygous. And here we're still going to use this probability, all right? Probability of getting this genotype. Can we do it? Equals, this thing's dying on, oh, here it is. Okay, probability of getting this genotype. What do? We can just do the events, right? We can do the events. Do the events out. We can draw the events out. Big G, big G, big W, little w. Okay. What's the probability of getting this event? Pulling a G. Pulling a G out of this cross. Big G, little g, big W, little w, big G, little w, big G, little g, big W, little w. What's the process again of pulling that G? 1/2, right? 1/2, 1/2, 1/2. Okay. Probability of all of those is equal to what? 1/16. Okay. That's the probability of getting this genotype, big G, big W, little w. What are we forgetting again? There's an order to it. Whenever you have these two here, because you can also get a big G, little w, big W. Whenever you have the heterozygous, you always have to remember to times it by 2, because there's 2 different ways of getting it. Okay, so 1/16 times 2 is equal to 2/16. over 16? 2 over 16. And that gives you what? 10 over 16. Okay.
Why do we multiply the 1/16 by 2 over here?
Over here, we multiply the 1/16 by 2. There's another way that you can do this. You can say, okay, here, let's look at it. Let's look at it in that cross, right? What's the probability in this cross of getting a little g, when you're crossing big G, little g, big G, little g? Do that little Punnett square, and what is it? 1/4, right? What's the probability of getting a big W, little w out of a big W, little w crossing with a big W, little w? 1/2, okay? So anytime you have a big W, little w, you have a heterozygous, you have to remember that there's an order to it. So you can either get a big W, little w, or a little w, big W. So this is just another way of looking at that. Or you can say you have to times it by 2 because in this, should have big G's here. Or you can calculate it the other way, going big G, big W, little w, big G, little w, big W. Each of these is 1/16, right? So that's where you times it by 2 because there's 2 different ways of getting it. We'll get more complicated with that stuff. Okay, any question? Some of you feel like you're understanding it, you're doing good. The best way to understand this and to really learn it is just do those problems, all right? So that's why I encourage you, and I know some of you, even this weekend, didn't do anything but problems. At least that's what you told me. And so I know you're in good shape. Okay, so here, right, this homozygous yellow or heterozygous, here's our 16th cell. What did we calculate using probability? 10 out of 16. Therefore, we should be able to count up the homozygous yellows, right? Where are the homozygous yellows? 1, 2, 3, 4, right? Heterozygous round, which was a heterozygous round? 5, 6, 7, 8, 9, and 10. Okay, 10 out of 16. There's our 10 out of 16. Which ones? Which are the ones that we had to minus out? The ones that were both homozygous round, homozygous yellow, heterozygous round. Where are those two? Right there. So those are the 2 16ths that we had to minus out. All right. Think about those. Think about that in terms of the space and the event space and things like that. And that's exactly where it comes from. Okay. So let's do a little more complicated one. Put this one on a piece of paper so I can look at it and decide just how hard I want to make the question on the exam. You guys I guess that's negative enforcement, right? It's like you'll purposely not do very well. Okay, so you're selfing. Let's not even worry about what these characters are. You have heterozygous big D, little d, heterozygous G, heterozygous W, heterozygous T. T is equal to terminal flowers. So yellow, round, tall, non-terminal flowers. All right. What's the probability in that next generation from that cross that you're going to get this genotype? Big D, little d, big G, little w, little w, little t, little t. Could you do a Punnett square?
Of course.
How big would the Punnett square be? How many cells? 16, 64, 256. Yeah, it would be huge. It would not be a very good way to do it. You right now can do it either of the two ways. You can do it using probability, or you can use it doing the fork line method. Phenotypes, I think, are easy fork line method. That's just me personally. Genotypes are usually easier, more straightforward, I think, using probability. Okay, I should pause this. Let's figure it out. All right, this could be a complicated problem, right? Lots of characters. What do we do when we have complicated problems? Make a big problem into one big problem into several small problems. Okay, so calculating the probability of this genotype, let's just ignore everything but the big D, little d. Okay, how hard is it to calculate the probability of getting a big D out of a cross between a big D little d and a big D little d? Can we do that? Can we do it in a Punnett square? If we drew out that Punnett square, what would the probability then be of getting a big D little d?
1 out of 2.
Maybe we'll even draw it down here. Big D little d, big G, little w, little t, little d. Crossing big D, little d, big D, little d. This first one, we'll do this, I guess. Here's a big D, little d, little simple Punnett square. What's the probability of getting a big D, little d out of this? 1, 2 out of 4, or 1/2. All right, did we see that? Okay, probability of getting a big D, little d, 1/2. Probability of getting a big G, little g out of this, a cross between a big G, little g and a big G, little g. Same thing, 1/2. Okay, probability of getting a little w, little w. 1/4. Probability of getting a little c, little c. 1/4. Probability of getting big D, little d, and big G, little g, and little w, and little t. Every time I said and, what do we do? Multiply. You multiply those out. What does that equal? 1 over 4, 16, 64. Okay, that's your probability. A lot quicker than drawing out a Punnett square. Anybody try to do it with the fourth line? Did it work?
Yeah.
All right, so we got a fork line. All right, so does everybody get that? So it's a lot easier if you just take little baby steps at a time. Just make 4 little simple problems, and at the end times them all together, and it ends up being pretty easy. Okay, so we can also do the same thing when we're looking at phenotypes. Okay, so in this example, we can say, okay, let's look at this cross between big A, little b, and another individual with a genotype big A, big B, little b, and let's look for the dominant and recessive phenotypes. Again, now we're sort of merging these methodologies, right? So here, we can say, okay, we're going to cross that, and we want to know the frequency of the dominant phenotype for both genes. Okay? If you remember before, when we're looking at phenotypes and it doesn't matter what the other allele is, how do we represent that? With a dash, right? And so here we say, okay, big A, big B, little b, big A, big B, little b. We're going to cross those. In the probability of the genotype, we're going to calculate the probability of getting a dominant phenotype, which is just a big A dash, right? And a dominant phenotype for B, which is just a big B dash. The dash can be a big A or little a, or it can be a big B or little b. It doesn't matter, right? Because the phenotype's going to be dominant. So again, we're breaking this down. Big A, little a, big A, little a. What's the probability of, let's just break it down here. This is a graphical representation. Same thing we're doing over here. What's the probability of getting a big A whatever in that Punnett square? So big A, little a, right? Do the Punnett square, big A, little a, little a. What's the probability of getting a big A or a big A, little a? 1. Does this one satisfy? Yes. Does that one satisfy? Yes. Does that one satisfy? Yes. It equals 3/4. 3/4 of your progeny are going to have that dominant phenotype, whatever that dominant phenotype is for A. What is the probability of getting, in that next generation, having the dominant B phenotype? Same thing, right? 3/4. Okay. Here, that's all that it's showing you here. See, we're making a little Punnett square out of it. What's the probability of getting big A, little a, or big A, whatever? 3/4. What's the probability of getting big B, whatever? 3/4. What's the probability then of getting big A, whatever, and little big A, whatever, and big B, whatever? Times those two together. What does that give us? 9/16. Dominant phenotype. Where else did we see this 9/16? On the Punnett square. What was the ratio? 9, 3, 3, 1. Okay, so let's go here, doing the same thing. Probability of getting little a, 1/4, right? Probability of getting a big B, whatever, 3/4. So recessive phenotype, dominant phenotype. Bring the 1/4 down, bring the 3/4 over. What does that equal? 3/16. The reverse here, dominant recessive, 3/16. recessive, recessive, 1/16. What does that give us? 9, 3, 3, 1 ratio. So all of these things sort of interact. Okay, we could draw it out here. Phenotype, frequencies dominant for both genes, right? So the probability of being dominant for both genes is 9 out of 16. The probability of being recessive for at least one gene, so you can be recessive for one gene. You can be recessive for A, you can be recessive for B, or you can be recessive for both of them. That's 9/17. Okay. So in a problem, you could have a problem that says, okay, what's the frequency in this cross? What's the frequency of the dominant phenotype for both genes? Or you could say, what's the frequency for being recessive for at least one gene? And you could just go through, use that methodology and calculate that. All right. That's the end of the probability. So, we have 15 minutes? Okay. So, we're going to go through and kind of go back to what we were looking at with Mendel when Mendel was looking at those ratios, that 9-3-3-1 ratio, and we were saying, okay, Mendel was close, but how close is close enough? Okay? How we do that, how we set this up in a more rigorous scientific fashion is to do hypothesis testing. So we sort of explicitly test hypotheses. How we perform that is through statistical tests. Okay? So we have our hypothesis. I don't have to define hypothesis for you probably. We collect our data. When we were collecting our data, we were collecting observations. Generally, most of the time we're collecting observations of phenotypes. So we go out into the garden and we look for tall yellow plants, short green plants, short tall green plants, and green, whatever the other combination was, right? And we score those if we were, say we were Mendel's helper, and we want to test to see if in fact our system is behaving close enough for us to say, yes, this gene or this character is the result of a single gene with two alleles, dominant recessive interaction between those alleles. Okay. So how do we do that? Let's go back now to the observed numbers in that Mendel study that we looked at before, and again calculate those expected numbers. Again, I want to reiterate though that these expected numbers are rounded. We don't want to round them. But we're just going to go back since we're familiar with this one and say, okay, we expected 313 round yellow in Mendel's experiment. How many did we get? 315. So close, but a little bit farther away. 104. How many do we get? 108, 104, 101. How close is close enough? How confident are we? That's when we want to do our statistical test. We can do this in real life. What if I would have flipped this coin up here? I would have flipped it, let's say 3 times, and I got all heads 3 times. And I said, this is a magical special nickel that it always does heads. How many? Or we say we did it 4 times and there were 4 heads. Would any of you? And I said, but I'm willing to let it go for $25. Who wants to buy this special nickel because 4 times in a row it got heads? How many of you would buy it? Probably none of you, right? So, but it's deviating from the expectation. But let's say that I did it, 1,000 times, and they were all heads. Then you might say, okay, yeah, there's a certain point where we deviate enough that we say, okay, something is fundamentally wrong, right? You're down in Wendover, you're playing blackjack, you lose 4 hands in a row. How many of you, after losing 4 hands in a row, and the dealer gets a 20, a 21, and a 20, and then another 21, and then the 5th time, she pulls up a 20, and you say, all right, that's it. There's something rigged here, and you tip over the table, and you march over to the pit boss, and you start pummeling him or something. How many of you would feel confident enough after 5 hands to do that? How about after 1,000 hands? Maybe after 1,000 you might do that, right? But probably wouldn't have any. You probably wouldn't have enough. Weaver State students would not have enough money to last 1,000 hands, or the Weaver State professors either, right? But if you did, you would think, okay, something's wrong. How do we gauge this? How do we quantify when something's a little wrong? Something about our fundamental assumptions about either the game, the flipping of the coin, or the way that these characters are being passed down from one generation to the next. How do we quantify that? And that's through statistical tests. Are there going to be deviations? Yes. What are causing these deviations? What are causing the deviations from? Are they mutations? I heard. What do you think? What are causing the deviations from between? Not deviations from the normal types, right? But deviations from expected numbers and observed numbers. What's causing the deviation for when I flip four heads instead of two? It's chance. It's random, stochastic, just sort of chance, right? So, do we always have random deviations within the system? Yes. What we have to do is quantify when it's more likely that it's just a random deviation or when it's more likely that something is fundamentally wrong. And we do that through this test. It's called, it's a part of a larger suite of tests called goodness of fit tests. It's called the chi-square test. So most of you have done a chi-square test. Most of you have seen this equation somewhere. What we do with the chi-square test, it comes from, this is the Greek symbol chi. Many of you have seen that before. It's the letter, what, in Greek? X or what? A lot of people think X. So, how many of you have ever seen, how many of you, those of you who come from, who have religious grandparents, right? How many of you have ever seen, in the holidays you say, Merry what? Xmas, right? And then what do your grandparents say? Don't say that, why? Am I the only one who had a religious grandparent? You're taking the Christ out of Christmas, right? Nobody's had a grandparent or somebody say that to you? Nobody? I had that. So it was very revealing when I learned the true meaning of this. So, chi-square test, what is this? Chi. This is chi. It's a Greek letter. Where does the word, what does the word Christus mean? Where does the word Christ come from? Is it Hebrew? Is it Aramaic? No. It's Greek. So it came into the New Testament as, a lot of the books in the New Testament were written in Greek. And so what is it really short for? What is it really? It's just a C. It's just a Greek C. So it's really short for Chimus or Christmas. It's just a short way of writing Christ. So next time your grandparent says that to you, tell them they're wrong and they need to go back to school. All right. So it's really chi. So that's where the chi comes. You've all seen that maybe, the chi, chimus, right? Okay, chi-square test. What we need when we're doing the chi-square test is we need observation data and we need to calculate expected data. And basically what we're doing is we're looking at the difference between our expected numbers and our observed numbers. We're squaring that difference because we don't care if the deviation, if we have fewer observed than expected or more observed than expected. To account for that, we square, and then we also want to scale it. We also want to scale our deviations. And so if it's a deviation of 1, if our expected number is 10,000 and we get 9,999, that's a deviation that's pretty small. If our expected number is 4 and we get a number 3, that's a much larger deviation. So we have to have a way of scaling that. That's how can we divide it by the expected. So chi-squared equals, what's this Greek letter? Sum, sigma, right? Sigma. Sum, it's the sum of all of the differences between your expected and your observed, squared, divided by the expected. So when we do a chi-square test, we first have to calculate expected numbers of, observations. We have to look at our observed number, right? We have to go through and get us one single, what we call test statistic from that. We then have to figure out degrees of freedom, and then we're going to compare our test statistic based on some sort of critical value that comes from a frequency distribution. So 5 minutes left, and I don't think I can get through all of the frequency distributions and everything else. So I think what will be better is if we stop there. and you just be familiar with the chi-square test. I'm not going to necessarily make you go through and calculate them yet because I want to take it through. I know a lot of you in 1110, don't you do Hardy-Weinberg? So I know a lot of you have done that, and my experience is students oftentimes leave there more confused than when they go into it, and so I usually try to kind of take a step back, and because it's pretty straightforward and simple if you do it. If you kind of go back and you go through it slowly. When we do the lab, sometimes it's a little quick, and we're calculating all these things, and I want to just make sure everybody is at the same pace. So let's stop here. Know what a chi-square test is, but don't go anything beyond this one. Question? Let me hear again. Degrees of freedom. Okay. Okay, so let's just go through and look at Mendel's dihybrid cross. Again, this is, oh, I got to pull this screen down. Again, this is, we've seen this cross a whole bunch of times, but now we're going to go back through. We're going to go back through and do the chi-square test. When we talked about it before, we said, okay, this seems to be really close. We'll count it good. Mendel did the same thing. And even some people have argued that it was a little bit too close, that he maybe threw out a few data or whatever. It was just almost too good. But Mendel did the same thing. This is close enough. This model, right? What was Mendel's simple model in this case? This model explained it, good enough. It was close enough. What was the model that he was saying is good enough? What is the assumption? The model assumption. In this case, with the yellow round, green wrinkled. If I had like an essay question, even though we didn't have any essay questions on that first exam, if I had an essay question that said, okay, based on this 9:3:3:1 ratio, it's really close, it's close enough, make you do a chi-square test, say it's close enough, okay, what does that tell us about our model? What is the assumption of the model with this, of this simple Mendelian model? So dominant recessive alleles at which genes? Both genes, right? So there's how many genes in here? This 9:3:3:1 first tells us how many genes? Two, right? How many alleles at each gene? Two. What is the interaction between the alleles? Dominant recessive. What else does it tell us about independent assortment? Yeah, that the genes are behaving independently too, or the alleles at those genes are behaving independently. Okay, so coming up with these results gives us a whole bunch of information, right, that we can say about the biological system. That's the whole sort of, that's the whole benefit of going through all of this. Okay, so back again. I know we've seen it a bunch of times, so I won't go through it. All of a sudden we get these are our results. 315 yellow round, 108 green round, 101 yellow round, and 32 green wrinkled. We see one phenotype that happens to be the same phenotype that you see in the F1, meaning it's a dominant phenotype, right? Happens to be the most frequent one, right? You have one that's the most frequent. You have two sort of intermediate ones, right, with somewhere around 100, and then you have one sort of rare that happens to be the recessive. So in general, the pattern seems to fit that what we know about that Mendelian system. But we need to test it. We need to do a goodness of fit test. This goodness of fit test is a chi-square test. And so generally when I do these, I like to organize it just like they do here in columns. Write down your phenotypic classes, right? Yellow round, green round, etc. Write down the observed number, right? Then put down the expected number. Again, I want to reiterate with this slide, don't round them up and down, even though they've done this. We're going to do it just because, even though they've done this in the book, I don't like to do it. You need to put to at least one decimal point, but preferably to 2 decimal points if that's what the results come out as you're expected. Okay, here this is not totally correct, all right, but that's okay. Get them in this column. Then you look for the difference of the two, observed minus expected minus observed. It doesn't really matter, right? Why? Because you're squaring it, right? So you find the difference between these two, square them, and divide it by the expected, and this is what you get. This now is a figure, is a number that tells you what? What does just this single one tell you? Is this the chi-square statistic? No, right? Because it's not summed, and that's in 1110. A lot of students get that confused. You have to add all of these up, but individually, each one of those, the 0.1, the 0.15, the 0.09, the 0.26, is telling you how much deviation is occurring in each of those phenotypic classes. So in a way, you're assessing which one is deviating more. So based on this, like based on these, right, these numbers, if you're just looking at these numbers without calculating this, could you tell me which one is deviating the most? Might be kind of hard, right? There's 2 differences here, there's 4 here, right? There's 3 here, and there's 3 here. You might say, oh, this one is deviating the most, right? But if you go through and calculate individually that expected, the difference between the expected and the observed square divided by the expected, you actually see that this one, this recessive is the one that is causing the most deviation. Why is it? Because it's being scaled by the expected number. And that helps to do that. So one important thing about the equation, the squaring of the differences eliminates, it gives you an absolute value of the deviation because you don't care if it's above or below, you're just worried about how much is it deviating, right? Each of those phenotypic classes, observed phenotypic classes from the expected, and then dividing it by the expected scales that particular one. So because the expected here is not very large, a deviation of just 3 is actually relatively larger than a deviation of, where's the other 3? Than a deviation of 3 here, because this expected is lower, okay? So you're looking at sort of the gross deviation of those. Does that make sense? Okay, that's sort of explaining to you the reasoning behind it. In a little bit more detail about that chi-squared. Okay, so let's look at another example. This is an example done around the turn of the century by a Dutch botanist Hugo de Vries. He was actually part of the group of geneticists that rediscovered Mendel and gave Mendel credit for coming up with this stuff, rather than just taking the credit for themselves. He, after rediscovering Mendel's papers, a whole bunch of them decided, oh, we got to go see, they're all excited and pumped up, and they wanted to go check out these Mendelian principles in other organisms and other systems. And so he was working with campions. He said, okay, I'm going to do just like Mendel did, and he looked at different characters within these flowers, these campions flowers. He looked at red foliage, hairy foliage, white flowers, smooth flowers, okay? He crossed them, right? This is kind of like what Mendel was doing with his pea plants. He crossed them, and all of the F1s were red flowers, hairy foliage, right? Everything's good. He's like, oh yeah, this is exactly kind of what I expected, right? What does that F1 tell you now? What information can you glean already from this experiment? Yeah, so red flowers are dominant and hairy flowers are dominant, right? What does he do? He lets the F1s, I don't think he's self-fertilized. He crosses all of the F1s, and what does he get out? How many phenotypic classes is he expecting? He's expecting 4. How many does he get out of this? 4, red hairy, white hairy, red smooth, white smooth, right? Dominant, dominant, recessive, dominant recessive, and recessive, recessive. Which is the most frequent class? The dominant, dominant. Is that what he expected? Yes. What is the least frequent class? Recessive, recessive. What are the 2 intermediate ones? The dominant, recessive, recessive, dominant. All right, so exactly sort of the general pattern that he was expecting, based on those Mendelian principles. So it's all good, publishes it, everything's great. He did not do a chi-square test at the time. He used this information, said, okay, this is great. This it furthers our proof of Mendelian principles, right? And everybody accepted it for the most part, right? If we look at these though, and we say, okay, let's now in retrospect, let's go through and do another chi-square test. How would you do it? So here you have your observed numbers. How do you get your expected numbers from those observed numbers? I'm going to cover it with the bottom here with my shadow. How do you do it? What's the first step? You have your observed numbers. What would you do? What would your first step? My back's hurting. What would your first step? What would your first step be when you're doing these problems? Remember, whenever we do these problems, we want to have a personal protocol, right? You want to say, okay, this is what I'm going to do when I see these chi-squares. What do? What did I tell you I do? I list them all in a column. Observe. List all of the, well, first I list all of the phenotypic categories in a column, right? And then I say, okay, the observed numbers in the column. After I list all those observed numbers, I then add them up. So I get my n value, your population size, right? N equals whatever those add up to. What does it happen to be here? 158. Then how do I get my expected ones? What's our expected ratios based on? Or what is our expected numbers based on? What ratio? 9331. 9/16 times 158 gives you this expected number. 1/16 times 158 gives you 29.6. 3/16 gives you the same. 1/16 times 158 equals 9.9. In this one, we're doing it right. We're keeping the decimals. Okay? Keep these decimals. We can go through and do a chi-square test with that. Here we see, okay, chi-squared. This in which one of these then is deviating the most from the expected? Do we know? Based on absolute values, right? Which one would you expect to be the one that's deviating the most? This one, raise your hand if you see dominant, dominant. We have a few hands. Recessive, dominant. I don't know, was it recessive? Yeah, recessive, dominant. Dominant, recessive. A few more hands. Recessive, recessive. A few hands. This one is a little bit harder to tell, right? Okay, so what would we do? We'd go through and we'd put into that equation. Observed minus expected squared. This total deviation gives us 4.02. This total deviation gives us 1.47, 9.09, 8.38. We now see that it was the red smooth category that was the biggest deviation. Add them all together, 22.94. Okay, is this close enough or not close enough? That's what we want to assess, right? This is just how you do the chi-squared. You know, how you get that test statistic. This is the first thing you need to do. You need to get that test statistic. Okay, what does this mean? What exactly does 22.94 compared to 0.51 with Mendel's? We know that this one obviously looks like it's way more out of whack, right? It deviated much more from the expected than the Mendelian one did, but what exactly is that telling us? Okay, we know what we're going to do, we're going to get our degrees of freedom, and then we're going to compare it to a value. But I want to go into exactly more background of where that stuff's coming from. Okay. So let's go back and look at Mendel's experiment. So when we're basically trying to answer the question, when do we know when a deviation is too big? Was 22 too big or not? What if that deviation, what if we added up our chi-squared and the test statistic was 7? Would 7 be big enough? Would that be too big or not? How are we assessing what is too big? And so that's what we're going to do. So if we went back, okay, never mind. So that's basically the question we're answering. But if we go back and we were to perform Mendel's experiment, so we know, okay, we say, all right, Mendel's was so close, we're confident that that system is operating under those assumptions. That there's 2 genes, 2 alleles in each gene, dominant, recessive, etc. Right? If we were to go back and do Mendel's experiment again, let's say, we wanted to do a nice under senior thesis or something, right? You said, I'm going to redo Mendel's experiments. And so you went and you redid it, and instead of getting Mendel's 0.51, you did it again, and you ended up getting, not quite these same numbers. Maybe you got a few less here, a few more here, 1 or 2 less here, and your chi-square statistic was 3. After you did that experiment, would you say, all right, it's time to we're going to publish this and just rock the genetics world. We're just going to we're going to throw Mendel under the bus, the bum. Would you be ready to say that? No, No, what would you want to do? You want to go recruit some other friends, right? And anyway, so then you say you recruit 1,000, all 1,000 of your Facebook friends, right? And you decide everybody's going to do it, and you did it, and somebody else did it, and all of a sudden they got 0.41, and then somebody else did it, and they got 5. Maybe somebody else did it. Is it possible that one of your 1,000 Facebook friends that did it would get a 20? What was the device? 22.5.
Yeah.
Is it possible that that could happen, even if the Mendelian system is all good? I mean, let's say no. Before we do that, we're going to go back and look at the research, and the research is solid. Yes, that's what that system is doing. With that system behaving exactly how it was supposed to, could you actually get results that were as deviating, as whacked out as De Vries in Mendel's system? Is it possible? Is it possible that you could flip a coin 20 times and get all heads?
Yes.
It's possible, right? So it's possible to get De Vries result even if we know our system is behaving the way that we think it should. So how do we assess that? If we went through and we got 1,000 of them, we'd say, okay, what we want to do is we want to plot out all of our results, all of our, Mendel face results. And we would plot them out, and this is what the plot would look like. We'd see here, the vast majority of them are less than, not the vast majority, but the biggest chunk of them are less than 1, less than 2. We just plotted them all out, plotted them all out, plotted them all out. Here is the one that was the equivalent of De Vries, and so this is continued all the way out here. But maybe there was one that did that. Maybe we did it 10,000 times. We now have what we call a frequency distribution. These are the results of all 1,000 or 10,000 of those experiments. Okay, so now we have to assess when is a deviation kind of strange. When is it too much, right? If we do it the original time. So let's say rather than like Mendel, we did it the original time when it was down here. Let's say in some experiment, like De Vries' experiment, we do it the original time and it's way out here. If it's the first time and it's way out here, this is probably what we call a rare event. So in statistics, anything less than 5% of the time is basically what we call a rare event. And so now we're sort of putting a line here. We're deciding, okay, this is where we're going to say, if when we do our experiment that first time, and our results are way out here in the hinterland, right? Way out on the edge, that fact is going to tell us, something ain't right. Something maybe is wrong with the assumptions of the system. And so, is this 5% sort of an arbitrary designation? Yeah, kind of, right? Could we say, okay, we're going to say something isn't right if it's at 10% over here, 10% or less. Let's say 10% is right here, or 20%, yeah, right here. We'll say, no, we're going to say something isn't right there. And so it kind of depends on the system that you're looking at. These different percentages, this 5%, this 10%, etc., those are what are your p-values. Those are your p-values that are telling you this is the probability of getting this result, even if our system is behaving like we're assuming it's behaving. So even though we know that in the case of Mendel's traits that it's 2 genes, etc., if we were to have gotten a result like De Vries way out here, and let's say that p-value was 0.00001, that's basically telling you, no, this is a result you would expect 1 in every 10,000 times. So it's pretty rare. That tells us, all right, we better go back and reassess what's going on. Okay, degrees of freedom play into this because the degrees of freedom will actually change the shape of that distribution. So that's what the degrees of freedom were doing. Okay, so looking at those two, here's Mendel's dihybrid cross, 0.51. De Vries, 22.9. Those are those results. Which ones are we going to reject? Here, because we have this critical value, 7.18, this is that 5% based on that distribution, that frequency distribution. De Vries is larger than that. So we reject the hypothesis. What is the hypothesis? We're rejecting that this system probably isn't a system that has two genes, two alleles at each gene, dominant recessive interaction between those genes, and that those genes or the alleles of those two genes are assorting independent. And then we go back through and say, okay, then what is going on? And that's what we're going to do too. But first we have to establish something isn't right, right? Something isn't right, let's reject that. Here we fail to reject it, or we accept the hypothesis. We never accept hypotheses, right? We just fail to reject them. We're saying, okay, this one is, we can use as saying supporting, yes, those assumptions are right. It is two genes, two alleles, dominant recessive, etc. Okay, so that critical value, where does that critical value come from? And so here we have, some of you who've taken statistics, you're not given sort of critical values that's easy. It's like, okay, here's your critical value. So if it's above this, we reject it. If it's below it, we don't. But you're given frequency distributions where the critical values, each column here has a set of critical values, depending on where you're going to set the line for this is a rare event. So in this case, we're looking at 5%, right? So we're saying 5%, therefore this is our critical value. Critical value of the P, if the P is less than 5%, we're going to reject it, okay? So each of these different lines, right, is representing different distributions that have slightly different shapes because they have different degrees of freedom. Okay, so here the degrees of freedom was 3 because we had 4 phenotypic classes, right? If we look at this, we see where was Mendel's 0.5, 0.51. So where is it on here? Like right here. So if we were to assign a p-value to that one result, that experiment that Mendel did, his p-value would have been 0.92. Okay, what does that tell us in more common language? That you should get that result how frequently? Generally about 92% of the time or so, right? If you're getting something that's going to happen about 92% of the time, is it time to panic and reassess your whole model? No, right? It's cool, everything's fine. Okay, now let's look at de Vries. Where was de Vries? All the way out here. Here's the lowest one here, 16. It's even more than that, right? So it's probably way out over, clear over here. This is maybe 0.00001, 1 in 10,000. So if the first time you do this experiment under these assumptions and you get something that's only going to happen, you would think has what kind of chance of happening, or what kind of probability of happening is 1 in 10,000. You think it's time to panic about your assumptions on your model? Yes. Yeah. So that's where you say, yes, this is whacked out. We need to reassess. Maybe there's more than 2 alleles. Maybe they're not dominant recessive. Maybe they're located on the same chromosome, so they're not assorting independent. So that's sort of the background behind the chi-square test. All right, makes sense? Questions? Okay, so here though is usually in like questions on the exams, I'll give you these critical values because it's easy, it doesn't take up so much space, right? And so usually you'll be given critical values. For our purposes, we're going to stick with that 5% critical value because that's how they do it in statistics. Sometimes in social sciences, I think they relax that a little bit because it's, you're dealing with humans a lot of times, so your sample sizes aren't big, and so you're just kind of looking for trends, and so they might relax it back to a p-value, a critical value of 20%. Sometimes when you're matching up sequences of DNA, right, and you're comparing them, you're saying, okay, I have this strand of 20 base pairs, and I want to match it up to 20 base pairs in the genome. Sometimes you're going to be much more strict with it. You're going to say, no, not 5% of the time, because I could randomly get that because there's over 3 billion possibilities out there. Randomly, it's a pretty good chance that this one will match up. My particular sequence will match up, and so maybe we're going to say our critical cutoff is 1% or 1/10 of 1%. But for this class and this purposes, we're just going to stick with 5%. Okay. So you can see that top part. I'll move that a little bit. Shape of the distribution, again, depends on the number of phenotypic classes. How we assess that is with degrees of freedom. Okay. So those are the key points. You can go back through. There are a number of examples of going through and doing chi-square tests in the back of the book. So I want you to go through and do them. You need to do them a couple of times, and after that, it'll be easy to you. Okay. All right. Questions?
So this end of chapter 3 will be on the new test.
Yeah. So basically, chi-square test, human genetics of chapter 3 will be on that next exam. Okay. Good question. All right. So now for the last part of this. Okay, so for the last part of this particular, of chapter 3, we do this, we like to do this whenever possible. We like to bring it home, right, and talk about human genetics. And so this last part, we're going to talk about Mendelian principles in human genetics. When we begin to try to assess human genetics or do analysis in human genetics, we see a little bit, we understand a little bit why it was so hard to come up with these general principles before Mendel, because basically we're trying to do it with humans a lot of times. Humans or other mammals, right? Cows, birds, chickens, whatever, right? They're a little bit more complex and maybe aren't as easy to work with. What some of the limitations of doing human genetics, and then even some of the limitations maybe of doing like genetics of cattle and things like that, is that the number of progeny is small compared to what Mendel was doing with his pea plants, comparing what DeVry was doing with his campions or whatever, right? They're a lot smaller. That is a limitation for us. Another limitation is we don't have a controlled environment like Mendel did, right? That's a big issue with a lot of human type studies, right? You see, do they still, in the Weaver newspaper and the signpost, do they have any ads in the back for, volunteering to be like human guinea pigs or not? When I was an undergrad, there were a lot of them down at the U of U. And you could actually go and make pretty good money, and I guess it's kind of taking advantage of undergrads, but I did one once, and I had to stay in the hospital for a week and 2 different weekends, right? And a lot of what we were doing up there, why they didn't just let us come in and say, okay, you're going to eat this food and let us go, right, is because it's an uncontrolled environment, right? So we had to actually go in this little corner of the U of U hospital, and we'd get these drugs, and then they draw our blood, and I had to stay in there for a week. Luckily, they had like a big collection of movies and a pool table and stuff like that. But, they wanted to control what we were getting, right? And then they would draw blood from it. It was over Christmas break. And so then this was a long time ago. And I earned like $1,500. So, that was a lot. That was back when tuition was like a fifth of what it is now. So that was good money, right? So there were a lot of people that did it, right? Well, a lot of desperate people that did it, like me. But even under this controlled environment, so we were shut into this corner of the hospital, this wing of the hospital, and they controlled what we were to eat, what we ate, and if they caught us eating something different, right, we'd be kicked out. So we could be there a whole week in the hospital and not get any money. But even with that, people would risk, every so often you'd smell like McDonald's in somewhere, right? And it would usually coming out of the bathroom or something, right? So people would smuggle in McDonald's and stuff like that. So it is hard to control the environment in human studies, right? You can imagine with if you wanted to do do Mendel's experiment, right, in human populations, it might be a little bit more difficult to get the F1s to mate with exactly who you want to mate with if you're dealing with human populations, right? We don't have those same liberties. And so there are a lot of shortcomings or a lot of obstacles when we're doing human genetics. what do? We have to just sort of look at human records. We have to look at genealogical records, right? A problem with looking at genealogical family records is one, that a lot of times they're incomplete. A lot of times we don't keep track of all of the kids, right? A lot of times we don't keep track of the conditions of all of the kids. A lot of times we think sometimes offspring of certain pairings are offspring of the pairings, but they really aren't, right? And so there's all of these things that we have to do we have to work through to do genetic analysis. But in spite of all of these obstacles, we've come up with some pretty good systems, and we've been able to describe some pretty good systems of human genetics. One of the top places for human genetics is just south of here in Salt Lake City. Why do you think the U of U has such a big human genetics program? Money. But to get the money, what do they have to? Where did it all start? Why out here in the sticks in Utah rather than Silicon Valley or somewhere? Why is it so disproportionate? One of the reasons is that if you go off of the U of U campus and you go down 3rd South, and then you get down close to the center of town, there's a little thing called the Genealogical Library. So what is in the LDS Genealogical Library? A whole bunch of pedigrees, right? And so to do human genetics, you need these family records, and then along with those family records, you then couple it with some of this other science that we're doing. But because of that, that's one of the reasons why the U is such a prominent human genetics place, is because they had really close access to, I think it's probably the biggest family record depository in the world. And so that's one of the reasons. Okay, so here we have a whole bunch of, traits that we've been able to figure out. Some of these traits. Here are some dominant traits that we'll deal with maybe a little bit. We'll deal more with these recessive traits in our problems, because those are the problems that are more interesting. But you have a whole bunch of dominant traits. You have dwarfism, you have short fingers, the brachydactyly. You have Marfan syndrome. Marfan syndrome is what some historians think Abraham Lincoln had. It's kind of this tall, gangly structure. Another one, Widow's Peak down here. You have Eddie Munster and Paul Ryan. I'm hoping Paul Ryan still stays prominent in politics, because otherwise I'm going to have to switch this file. So I'm cheering for him, right? That's Widow's Peak. That's that sort of widows. That's sort of sort of spike in the middle of your brow, right? That's, we know, is a dominant trait. We also have been able to identify a whole bunch of recessive traits. These recessive traits we're going to deal with in a lot more problems. Albinism, ketonuria, phenylketonuria down here, sickle cell anemia. These are recessive traits that we've been able to assess based on looking at pedigrees and things like that. Okay, so that's what we're going to do now. We're going to start looking at pedigrees. Okay, so in order to look at the pedigrees and get information on the pedigrees, we have to establish some, some sort of way of communicating these things, some sort of conventional way that you can see it on a problem, and you'll know, okay, this is what this problem is telling us. This is what this pedigree is telling us. This sex unspecified, we don't really run into that too much. We usually, are pretty clear about which are the females, which are the males. The circles are the females in these pedigrees. The squares are the males. We fill in the circles and the squares to indicate that the individuals have the trait. Sometimes we have the line through it means they're deceased. Sometimes because of spacing in the pedigrees, rather than drawing out, so say this is a family of 7 with 3 girls and 4 boys, we would draw it out like this rather than drawing it all out like that, okay? The connections, the lines between the males and the females means that there's mating going on. What's that?
So, in questions like that, where it'll say there's a family of 7, like on the questions, we turned in the added question was, it said, I think, in a family of 5, what are the probability of having 3 girls? If it says a family of 5, are we to consider 2 parents and 3 kids, or?
No, not usually. Not well. Yeah, Not usually, but it usually should. Did I not specify? I usually should say 5 kids. Yeah, usually don't count the parents in that. In that one, yeah, I think in the, is it one? It's probably one that I wrote, right?
I think so.
Yeah,
Last.
we'll get to that though. That's when we start talking about the binomial here in a second. We'll get to those. And yeah, it should be specified. And if it wasn't specified on the exam, then that would be something like, oh, I calculated. But it should say in a family of 5 kids. Yeah. Or in a family where the total number of members equals 10. Maybe you could. That would be where you would. But generally, I don't. I try. The number I give is generally the number of kids.
Okay.
Okay. So here, this is what the pedigrees look like. So here you have a male and a female. They mate, and how many kids do they have? 4. 1, 2, 3, 4. So you draw it here, you bring it out like that, bring it down, bring it down, bring it down. How many girls, how many boys? 2 girls, 2 boys. How many people affected?
1.
Is it a male or a female?
Female.
Yeah, see, you guys got it. This is easy, right? The Roman numerals usually indicate the generations. The Arabic numerals indicate the individuals within that generation. Okay, so pretty simple. How many of you have seen these kind of pedigrees before? Probably somewhere. Yeah, it's not too. Reading the pedigrees usually isn't, that big that too much strain for this. Okay, what we can do with these pedigrees though, is we can look for patterns. When we look for patterns in pedigrees, we could say, let's just go back to the simple. What does a dominant trait look like compared to a recessive trait? Here, this is a pedigree. How many generations? 1, 2, 3, 4, right? If we're looking at this pedigree, what is the relationship between this person and this person? Yeah, first cousins, right? What is this individual? This is somebody who's just from outside, from some other pedigree that's coming in and marrying, this guy's marrying this female. We're going to make some assumptions about people from the outside. When we're looking at, I'll talk about it in a second. Okay, so what is it? How do we know that this is a dominant trait?
It doesn't skip a generation.
Yeah, if it was recessive, it could skip a generation, or basically you could have parents that don't have that trait. Here, if it's a dominant trait, you see it in every single generation. Not only that, but everybody who has it has a parent that has it, right? A lot of times, if that dominant trait in the homozygous form is deleterious, we can assume that the dominant trait is present in the heterozygous form. Okay, but you'd have to be told that in the problem. Okay, what then does a recessive trait look like? Here's a recessive trait. Recessive traits, many times, not all the time, but many times, as in here, right? You have parents that don't have the trait. Then all of a sudden, the recessive condition ends up here. What does that tell us about the parents? They're carriers. What else are they, relationship-wise? First cousins. So what does this mean? This means there's a first cousins mating. They have 3 kids. One of the kids has this condition. Okay. What else do we see? We're going to talk a lot about inbreeding. What else can you tell us about this pedigree? You're going to be given these pedigrees, and the trick is to be able to look at these patterns and then start to generate information based on this. Can you follow this allele through the different generations? If this individual has it, what do you know about these two? Let's just say it's albinism, little a. What do you know about the genotype here? You want to take these pedigrees and figure out genotypes from these pedigrees. What do you know about this genotype here? What is it? Little a, right? Okay, what then are these 2 genotypes? Heterozygous, big A, little a. Okay. Here, then, do you know this individual? Do you know this genotype? Do you know this genotype? There's a little a somewhere, right? And so here, this is big A, little a. This is big A, little a. Then what would it likely be? Where does this little a come from? This little a, one of the little a's. One of the little a's was here, right? One is here. One probably then came from where? had to come from the mom, right? Because with recessive, what I was mentioning before, with the recessive conditions, we assume that they're rare. So unless there's contrary information, somebody that's coming into this family and mating from the outside, we're going to assume it's big A, big A. Okay, unless there's contrary information. If this individual had albinism, was an albino, would that be contrary information? Yeah, that would then mean that this guy that's coming in also was a carrier. And so what you'll do is you'll go back, trace that back, and try to figure out exactly what the genotypes are, and then begin to make some predictions. And we'll do that probably not today, but in a little bit. Okay, so pedigrees. Pedigrees help us to overcome these obstacles of human genetics. Okay, another obstacle of human genetics is, like we mentioned before, that humans don't have, a lot of kids. They don't know that you can't have 100 F1s from one mating like you could possibly in Mendel's pea plants. Since we have a much more difficult time of generating the offspring, we aren't able to address these situations with as much statistical power as we would if we were doing it with pea plants. But one sort of saving grace of that is that a lot of the things that we're looking at in these problems is conditions that have one of two states. So maybe we're looking at male versus female, or maybe we're looking at affected or not affected, albino versus non-albino, right? Colorblind versus not colorblind, or things like that. Or we're working with dominant recessive phenotypes. And so there's one of two states. Because there's one of two states, we can approximate these things by what's called a bi meaning two-state distribution, a binomial distribution. So for instance, so now we're getting to those problems. A couple, both carriers for albinism, have 4 kids. How many of their offspring are going to be affected? This is a typical problem that you could have to do. So what would you do in this case? And again, we want to establish some sort of personal protocol about how you're going to do these problems. Okay. What I like to do is I like to say, okay, I'm going to translate the information here into something that's a little bit more organized, right? So a couple, right? So here's a couple. I could even do it like pedigree form, right? If I wanted to. A couple, both are carriers for albinism. Albinism is what kind of recessive trait, right? So they're both carriers, so we'll just call them big A, little a. Big A, little a. So I'm getting away from the pedigree now. They mate and they have 4 kids. How many kids are going to be affected? So what I want to do, I want to go back to what we did earlier in the chapter and go back to the probability stuff, and I say, okay, having 4 kids is what? It's a process, right? How many events of the process of having a kid, how many outcomes or events come out of that? 4, right? So I go here, okay, 4 outcomes, right? So what is the probability of, if you have 4 kids, what's the probability of the first kid being affected? Okay, so we'll say, well, actually this is I'm kind of getting ahead of myself. If you had, what is the probability of 1 out of 4? Okay, what is the probability? We could do a Punnett square, right? And if they go big A, big A, big A, little a, big A, little a, little a, little a, right? Okay, so 1 out of 4 offspring, you would expect one of the 4 offspring to be albino, right? Okay, so would that be the answer to that? Do you know? Is it for certain? Is it possible that none of the offspring are albinos?
Yes.
It's possible, right? So now I should have done that first. So let's say, okay, then what is the probability that none of the offspring are going to be albinos? You have 4 events, right? You know, because that's what we'd like, right? We don't have to deal with the cost of, you know, of dealing with a kid that's albino, protecting them from the sun and things like that. So what is that probability? Here's one event, another event, another event. What's the probability that that first kid is not going to be albino? Go back to that Punnett square, it's 3/4. Okay. What's the probability of the second kid? Same, 3/4, 3/4, 3/4. What's the probability that this event and this event and this event and that event occur? Multiply. You multiply 3/4 raised to the 4th power, and that equals what? To it. It's in the next slide since I'm okay. So we go, oh, it actually isn't in the next slide. Well, maybe we should time that. 3, 9, 27. 27 times 3 is what? 81 over 4, 16, 64, 256. Yeah, so it's 81 over 256. Okay. So 81 over 256 is somewhere between 0 and 0.1%. And so here, that's our probability. So is it likely that you're going to have 4 kids that are unaffected? Oh no, these are affected, sorry. So it's actually 0. So, oh, it's actually over here. Sorry, I did the wrong one. So what's the probability that 0 of the kids will be affected or all of them will be unaffected? It's somewhere around 30%. So it's a pretty good chance. Here, what's the probability that all 4 kids are going to be albino or affected? You do 1/4 raised to the 4th power, right? Over here. The point is that you can calculate the probability of each of these. What is the highest probability then? That how many kids will have it? What are you most likely to have? It's what scenario you're most likely to have in this situation? 1. It's a, you know, just slightly between 40 and 50% chance that if this couple has 4 kids, they're going to have at least one albino kid. Okay, so that's the probability distribution, or that's the binomial distribution. Okay, so we have what, 2 minutes? I always like to end it with a problem. I want you to try to figure this out. Okay, in a family of 3 children, what is the probability that they will have all girls? Again, it's binomial. Why? We're dealing with 2 different genders, right? Family of 3, what's the probability that they'll have all girls? What's the probability they'll have a girl and then 2 boys? What's the probability they'll have 2 boys and then a girl? What's the probability they will have all boys? Okay, how many events is there? 3, right? 1, 2, 3, 1, 2, 3. Draw out the 3 events for each of these. All girls, G, G, G. 1 girl, 2 boys, right? 2 boys. Oh, 2 boys, and then 1 girl, right? And then all boys. How are we going to do this? What's the probability of having a girl? Everybody knows, right? 1/2. 1/2, 1/2. So in a family of 3, having 3 girls, what's the probability of having that? All 3 being girls? 1 in 8, 1/8, right? What's the probability here? All 1/2, right? 1/8. What's the probability here? 1/8. What's the probability here? 1/8, right? So add those up. 4 8s. We're missing some, right? What are we missing? Which are we missing? I actually think I wrote this problem. This should be, let's do, instead of 2 boys and 1 girl, let's go 2 girls, let's say this is 2 girls and 1 boy. So 2 girls and 1 boy. These are the combinations. This would still be 1/8, but we're missing something. What are we missing? Anybody know? Why isn't it adding to 1? Order, I heard. Right? So what is it? Well, which one is it? Are we missing order? Is there an order to this one? No, because they're all girls. Is there an order to this one? No. Is there an order to this one?
Yes.
Yes. You could have a girl, boy, girl, or you could go girl, oh wait, sorry. Or you could go boy, right? Girl, boy, or you could go what? Boy, boy, girl. So we have to consider the order. Each of these is also going to be 1/8, 1/8. With this one in the order, you to do boys and girls. I mean, to do the orders. Okay, so to go home, this next, I screwed that up. Okay, so now let's go to a situation that's a little bit more complicated. This time, rather than stop talking about girls and boys, we're going to talk about some parents that are carrying some recessive condition. We'll call it color blindness or albinism, whatever we want to call it. Well, it's C, so we'll say color blindness. Okay, so you have two parents that are carriers, right? Big C, little c, big C, little c. They have offspring. They want to know what is the probability that they will have, you know, what are all these different probabilities of of them having all their kids that are colorblind, none of their kids that are colorblind, 2 of their kids that are colorblind, etc., etc. Okay, so here we have this situation set up. So write it down, because then we're going to go to the next slide and you're going to have to try to calculate those things. So remember, what are we always doing? Okay, we write down all of our initial stuff. So we have big C, little c, mating with big C, little c, and then we have 4 children. So right away, let's just go over here and start 1, 2, 3, 4 events. Okay. So now, what are the things that we're going to answer? What are the questions that we're going to answer? Okay, let's go with the first. Let's go with the first one. What is the probability of none being affected? So here you have your kids, 1, 2, 3, 4 kids. So this one is unaffected, so I just put a little U under that event, right? Here another U, another U, and another U, all under the hash marks, so unaffected. Okay, so in this mating, it's an easy, we've done this a whole bunch of times. Anybody still need to do the Punnett square with this? If you do, don't feel bad. It's better to be slow and accurate than quick and not accurate, right? On the exams, you have as much time as you want taking them, so do it however you feel the most comfortable. Okay, so what is it? What's the probability of being unaffected? You can either be big C, or big C, little c. So it's 3/4, right? 3/4, 3/4, and 3/4. Okay, so this one, and this one, and this one, and this one, you multiply, right? What does that equal? Somebody with a calculator. Elizabeth, you have a calculator? You want to? 81 over 256. Okay, does everybody see that one? All right, now let's everybody go through and do together, what is the probability of them all being affected? This is going to be, we could draw it out the same way. Instead of a U, we'd have an A. Instead of 3/4, we'd have what? 1/4. 1/4. And so 1/4 times 1/4 times 1/4 times 1/4, same thing as 1/4 to the 4th. And what does that equal? 1 over 256. So there's a really, really low probability with a family of 4 that they are going to be unaffected. Or, I mean, they are all going to be affected, sorry. All right, so now I want you to do the other 3 problems. What is the probability of 1 being affected? Just 1 of the 4. What is the probability of 2 of the 4 being affected? And what is the probability of 3 of the 4 being affected? Now it gets a little bit more complicated. Okay, so we'll take like 2 minutes. Well, we'll take whatever. You guys need to at least tackle the first one and maybe the second one. Okay, let's do that first. Well, not the first one, the third one. Okay, what is the probability of 1 being affected? So here up on the board, I draw 4 hash marks, right? Underneath I put a big A for affected, big U, big U, big U, 4 different events, right? What is the probability of this first one being affected? 1/4. Does everybody see that? Based on Punnett square. What's the probability of the second one being unaffected? 3/4, and then 3/4, and then 3/4. So what's the probability of the first one being affected, and the second one being unaffected, and the third one being unaffected, and the fourth one being unaffected? We multiply, right? Because I just said and 3 times. So what does that get? What does that get us? 1/4 times 3/4 times 3/4 times 3/4 is again over 256. 27, right? 27 over 256. How many of you got that as your answer? Okay, how many of you didn't get this as your answer? All right. So, what is this question asking you? What is the probability of one being affected? So this is a probability of one being affected if it's that first one. But what else do we have to do? We have to do the different combinations, right? And so let's say the first oldest child was unaffected, then the next one was affected, and then unaffected, unaffected. What's the probability of that particular one happening? Same thing, right? Carl said, what'd you say? Say it louder, because I erased it. 27 over 256. Okay? Probability of that one is 27 over 27 over 256. That's the first one. Is there any other combination? Yep, you can have unaffected, the oldest, the second oldest unaffected, the third oldest affected, and the youngest unaffected. What's the probability of that one? 27 over 256, right? Well, is there one last combination? Yep, you can have the oldest, the second oldest, and the third oldest being unaffected, and the youngest being affected. That is 27 over 256, right? So the probability of the answer to this third question, what is the probability of one being affected? It could be this one, or this combination, or this combination, or that combination. So what do? You add them all up together. Did anybody do that? 108, is that what it is? Okay, so the total is 108 over 256. Okay, that would be the answer to that. Could you then do this same one here, this last one? What would change doing this last one compared to this one? You're still going to have how many different combinations? 4, right? So the last one, the fifth one, not the fourth one. So we skip this fourth one. So probability of 3 being affected, right? So instead you're going to have big A, big A, big A, U. You're still going to have 4 different combinations, right? But the probability of this first one is going to be 1/4 times 1/4 times 1/4 times 3/4, right? That equals what? 3 over 256. But there's going to be 4 of those combinations, so you just times 4 by 3 over 256. And that's going to be equal to 12 over 256. Okay. That's that very last one. Okay. What's that? 12.
It's 4 over 1
Because
times
it's
3
4.
over
4 over 1. I know. How long has it been since you all have done fractions? A while. I know. That actually is not an insignificant thing when you start doing these, because you got to remember how to do all this fraction stuff. Okay. So that one is pretty much the fifth one here. The fifth problem here is pretty much like the third problem. You just have to change those individual probabilities. This one, though, the fourth one, is unique in and of itself. So how many of you have done this fourth one? All right, let's spend, now that you kind of saw how to do the third one and the fifth one, let's take maybe a minute or two and try to do that fourth one. Okay, so show of hands, how many have done that one? Okay, so most of you, so what was the difference to it? Well, those of you who haven't done it, where are you hung up? Usually, this one is a little bit more complicated. Why? Or maybe somebody who's done it, tell us why it's a little more complicated.
There's more permutations.
Yeah, so there's more combos, or there's more permutations, right? So, what is the probability of what? What is the probability of 2 being affected? So you can still go. You can still go 1, 2, 3, 4, affected, affected, unaffected, unaffected, and you could draw these out, right? You could draw out all 6. You can say, okay, affected, unaffected, affected, etc., affected, unaffected, affected. I'm writing something that's saying completely opposite. Affected, unaffected, unaffected, and affected, right? And you could go do all those different combinations, right? How many combinations were there? 6. Okay, so this ended up being 6, so then you go 6 times. What's the probability of let's just look at this top one, of the first one being unaffected? 1/4. 1/4. Second one? 1/4. Third one? 3/4. Fourth one? 3/4. All of them, 9 over 256, right? times 9 over 256, 6 different times because there's 6 combinations. That equals 54 over 256, right? Yep. Okay, so you just need to remember when the order doesn't matter, right? You're not asking a question of this very, very specific order, that you need to figure out all of the different combinations. Okay, so here this slide this slide shows you all basically what we just did. Okay, if you were to draw out the 2 permutations, or the permutations for the 2 affected, 2 unaffected, you would get 6. However, sometimes if we were to have, let's say, 5 kids or 7 kids, then drawing out the permutations becomes much more difficult. For instance, in the case of, let's just take an example and just think about it, and then we'll do the permutation formula. But let's say you had 7 kids, 1, 2, 3, 4, 5, 6, 7. Anybody come from a family of 7 kids? Am I the only one? 7 and a half, actually. I have a half brother. So 7 kids, 3 boys, 4 girls. So the oldest, I have an older brother, a sister, then me, right here with the asterisk, then another brother, then another brother, all 3 years apart. That was pretty brutal for my mom. And then 2 younger sisters. Okay, so what is the probability, because I know you all want to be like me, of when you have your families, of having a family of, what is it, 4 boys? 4 boys and 3 girls. Okay, how many different permutations are these? You'd have to go through and say, okay, boy, 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 girl, 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 right? And then you have to go boy, boy, start distributing through, girl, boy, girl, girl, boy, 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 girl, girl, boy, girl. Pretty soon, you're going to get confused, right? And so, fortunately, we have a formula, which is the formula for permutations or combinations, that we can just plug them in. All we need to know is how many different events, right? Up here on the top, that is what we're going to call n, and then we have to know the number of each of the classes. In this case, it would be the number of girls versus the number of boys. Okay? So, some of you, I'm not sure which class, maybe it's 1030 you do permutations. Is that true?
1040.
What's 1040?
Stats.
Stats, you do permutations. Maybe not 1050, right? Just an algebra, but maybe you have, maybe you've done them a little bit in high school, whatever. Some of you probably, it's been a while, so let's just go through what this formula means. Okay, so n is the number of events, right? So you have n with the explanation mark. That explanation mark means factorial. Okay, so in this case, well, let's just draw out the whole formula. Okay, x factorial over y factorial. N factorial in the case of my family is what? N is equal to what? 7. So 7 factorial is just the same thing as writing out 7 times 6 times 5 times 4 times 3 times 2, and then you can't do times 1, you don't have to change anything, times 1. Okay? So that's what factorial means. So here we have 7 factorial over, in my case, we'll call x the females, right? So the x, the females are 3 factorial, right? 4 factorial. So if we were to draw that out, I'll have to write it a little bit small. 3 factorial is equal to what? 3 times 2 times 1. And then this whole thing is we're going to times. So 3 factorial times 4 factorial. What's 4 factorial? 4 times 3 times 2 times 1, right? What do we see? Since this is a fraction, they cancel out. So this one, right? 4 times 3 times 2 times 1. 4 times 3 times 2 times 1. Those cancel out. And this reduces to 7 times 6 times 5 on the top. On the bottom, 3 times 2 times 1, that equals, we can just do it out, right? 6. So many times 7 times 6 times 5, 42, 2, 10, 2 something. 42, times 5, what is that?
The six-
Oh yeah, or you can, oh yeah, good. We can just keep canceling. What does it equal? 35. Good, Elizabeth. We can even continue to cancel. We don't have to do anything. Okay, so 35. So what does that mean? There are 35 combinations, different combinations of getting, in a family of 7, of having 4 boys and 3 girls. Okay, so if you want to, go back and do the factorial for that last problem, the middle one. The middle one was 4 kids, right? How many, and 2 affected, 2 unaffected? That was it, right? So go back and just do the factorial problem for that one and see if you come up with the same. If you don't, then do it again. So I'll give you like a minute to do that. Okay, so having confirmed that we did it right in the time before, one last thing that I have to mention about factorials is sometimes you can have, say for instance, you wanted to figure out in a family of 7, right? How many kids? So let's say 7n again. What's the probability of having 7 boys and 0 girls? You would go through and you say, okay, 0 factorial times 7 factorial. Okay, we have to define 0 factorial. 0 factorial is not 0 times 1 or whatever, right? 0 factorial is what? 1. So it equals 1, and that makes sense because if this equals 1, right, 7 factorial, 7 factorial, 7 factorial over 7 factorial equals 1. Therefore, that tells us there's only one way to get all 7 kids being males, or only one way of having all 7 girls be females. So you could actually do the factorial on all of the problems, right? It's consistent throughout. Okay, everybody good with factorials? If you haven't done factorials in a while, go through and do some of these. Okay, so what is the probability of having a family with 7 children, having 5 boys and 2 girls? You could go through and do that the same way. We're not going to spend time because we just kind of did that one. This is one just giving you a problem that you can go through and sort of do on your own. Okay. Here, another one. Oh yeah, another problem you can go through and do on your own. Does anybody remember the Waltons? Kevin? All right, I needed you. All right, Kevin remembers the Waltons. So the Waltons had 7 kids. How many boys? John Boy, Jason, Aaron. Yeah, that was a girl. Benjamin, James. 4 boys, 3 girls. Okay, what's the probability of having a family? Say you idolized the Waltons and you wanted a family just like the Waltons. What are the probability of you having a family exactly like that? Could you calculate that? Go home and do it. I just want to give you a couple of problems you can do. Seriously, you guys never heard of the Waltons?
I have, but it's only because of Family Guy, so it's not really.
Oh, on Family Guy? Okay, question.
So I got a question on the one we did that had the 6 different possibilities. What is the probability of 2 being affected? So when I'm doing that in the factorial way.
Okay, so we're doing 6. So, okay, we'll go back and do it. So it was 6. That was the comment. How many different events?
6.
We're looking at 6 events and having 2 or 4 events.
4 events.
Okay, yeah, maybe that's what.
Well,
So 4 events.
So it'd be 4 factorial.
4 events. So 4 factorial on top. On the bottom, how many were we asking? 2 affected, 2 unaffected?
So that would the x be 2 and y be 2?
Yeah, so the x would be 2 and the y would be 2. 2 affected, 2 unaffected. Okay.
That comes out to be 6 though, right?
Yeah. So if you go through and you do that, that equals 6. How many different combinations were there? Do you remember?
6.
Yep. You got it.
Well, but the correct answer was 54 over 256, wasn't it?
That's the
That's-
answer. But the
yeah.
number of permutations was 6. The number of combinations
Yeah.
was 6.
Oh,
So you had to do
that's all-
6
That's all we're.
times.
Just give me what you would times the answer by.
Yeah. That's so what was it? What was the original of just getting 2 and 2? It was 9 over 256. Is that right? So that's that part. Yeah. So it's a 2-part thing. You have to calculate. Okay, what? And you can choose any of the combinations you want, but you have to figure out. Okay, when I get 2 affected and 2 unaffected, regardless of what the order is, what's that probability? And then you have to times it by each of those different orders.
Okay.
Okay. So that's what you would do. Okay, so I think the biggest thing to remember here is when to use the permutation formula and when not to. And in reality, you could use a permutation formula every time. Sometimes it'll just cancel to, it'll just reduce to 1. All right.
So the whole idea of that is telling you how many possibilities.
Yeah, it's a shortcut. Rather than having to draw them all out on the board, you know, the 2 affected, 2 unaffected, all 6 times, you could just go back, use this formula, and calculate it. And this formula is simple enough that it won't give it to you on the exam. You just have to remember, okay, number. It's just, and that's it. That's a good formula to kind of know in other situations too. Whenever you have to figure out, okay, what are the possible combinations, you can do it. Okay, Dan, you had a question?
What, just on the Waltons, what's the numbers referring to?
Those numbers are referring to different subsections of Wikipedia. So if you click on John Boy Walton, you'll get to know more about John Boy Walton. So I just copied it. I have to confess, I don't know off the top of my head all of the Walton kids by name. I remember John Boy. So incidentally, on Family Guy, what did they do? They did the "Good Night, John Boy" thing. Yeah, that's what. That's where the Waltons live. Yeah, it's exact. So that's how they would end each Waltons episode. Okay, so it made its way into popular culture. That's good. Okay. All right. So now let's go back and kind of merge 2 of the things that we've already learned about. Okay, we learned about the pedigrees, right? We can understand, we can read these pedigrees. We've also learned about probability. So let's combine those 2 things to figure out something that maybe if you were to become a genetic counselor, that you would have a client come in and ask you. Is anybody interested in becoming a genetic counselor? No? They make more money. They make more money than professors. Are you sure you don't want to? You all know that that's still not very much, right? No, actually, I think they make like 120, 130,000 a year. No big loan for med school. Still, nobody wants to be a genetic counselor.
So what are the limitations of that? I mean, you go to a genetic counselor, and at what point are they? Because I mean, really, we have the capability to do a lot of things that ethically people aren't allowing right now, right? So you go to a genetic counselor, and
Are you talking about like designer babies type stuff?
yeah, I mean, at what point do they say, sorry, we won't do that? You know what I mean?
Well, see, so the genetic counselor, mainly what they're doing is they're like a situation like this. Say 2 people come in, they have albinism in their family, and they're wondering what their probability is of them having kids that maybe would be affected, right? Or you have a certain allele, you test it. What genetic counseling is going to become more of is testing positive for a certain allele and then really translating what that means at the ground level for people who have just tested positive, say the BRCA1 allele that we talked about, the breast cancer allele. Okay, what does that mean? You're going to now have to make a decision, do you want to have a mastectomy or not? You know, and so then the genetic counselor brings them in and sort of breaks down, okay, this is the probability, because there's a lot of misinformation out there about exactly, is it, you know, people will get test positive for some allele and they'll think, oh, for sure I'm going to get it, right? Yeah. That's not necessarily true. And so what genetic counselors do is work you through that. So I'm sure there's, you know, in a genetic counseling degree, master's degree or whatever, I'm sure there's some psychology involved, there's some statistics involved. You know, I don't think it'd be that bad a gig. Okay, so before you decide to become a genetic counselor, maybe let's do one problem to see how good you are at it. Okay, so here you go. Here's a pedigree. You have 2 people come in, right? And they say, let's say these 2 people, R and S, they come in. And R says, you know, I have siblings in my family. I have 2 brothers that are albino. Neither of my parents are albino. And 3 of my other brothers, all boys, 3 of my other brothers are non-albinos, okay? But I married an albino. So I want to know what is the risk that first child is going to be affected with albinism. Can you calculate that? Here you go. There's a whole bunch of information up here, right? Here's our pedigree. Try to calculate the pedigree. Come up with a probability for T having albinism. All right, just take a minute to do that. Okay, so whenever we're doing these pedigree problems, my suggestion is that the first thing that you do is you write on the pedigrees the genotypes that you know, okay, based on the pedigree. So, based on the pedigree, we know that the two brothers, right, are little a, right? Little a. Why? Because they're albinos, and we know that it's a recessive condition, so they have to be little a, right? Who else do we know is little a? The wife, little a. Okay, because, this is what Christian was asking, because we have an offspring that is little a, what do we know about the genotypes of the grandparents?
Heterozygous.
They're heterozygous, right? They have to be big A, little a, big A, little a. Do we know now the genotype of this individual?
No.
Not necessarily, right? It could be big A, little a, or it could be big A. So therefore, just like this one, there's brother, same sort of thing. We don't necessarily know which of the genotypes, which of these two genotypes that the father right there is, right? So what's the probability that that person right here, R, is a carrier? Okay, so if we do a Punnett square, right? We say, okay, here's our Punnett square, big A, little a, big A, little a. We do it through, right? And we say, okay, 2 of the 4 cells are carriers, right? So that's when we say 1/2. But soon after we see that, we think, wait a second though. Do we know that that individual isn't one of these genotypes? Yeah, we know that R is not little a. Why?
Because he doesn't.
Because he's not albino, right? So then we sort of just X that one out. So really, the probability, we'll write it as probability that R is a carrier is equal to what then? Yeah, 2/3 is 2/3. Okay. Now, let's just say given R is a carrier, so we're not going to write this in, maybe we'll write it in parentheses. So if R was a carrier, then what is the probability that T is going to have albinism? 1 out of 2. Does everybody see that? Should I draw up the Punnett square? No. Okay. So probability given, we'll even draw it out here. Given R is a carrier, well, this is a long sentence, never mind. We'll just say, what is the probability? We'll just say, what is the probability? We'll just, in our mind, remember, okay, R has to be a carrier. Probability child's affected is equal to 1/2. Okay, these are 2 different events, right? In order for a child to be albino, R has to be a carrier, right? That's that probability, 2/3. And both R and S have to pass down the little a allele. That is equal to 1/2. If it has to be this event and this event have to occur, what's the total probability? How do we figure it out? When we hear and, what do? We multiply. You multiply those 2. What does it equal? 2/6 or 1/3. So that's the probability. All right, how many of you got that right? How many of you got it wrong? The rest of you? How many of you didn't get it, but now you can do it?
I have a question though.
Question.
If he's heterozygous dominant, AA, he won't have.
Well, if he's AA, he's homozygous.
Homozygous
Uh-huh.
AA, then if his
Then the
gametes,
probability is
he won't pass it on though.
yeah,
So,
exactly.
but
Then the
then-
probability would be 0.
but then how does that factor into the 2/3?
That's not part of that. So you could make another question. You could say, okay, what is the probability that the probability, it's actually a little bit more, now this makes me mom. I was going to say, what is the probability that none of the children are, or that your first child is not affected? And so you could do that, and if you did that, you would have to factor in the one-third of the time, none of them, plus then the combination one you'd add to that, so it's a little more complicated. But yeah, in this case, by this 2/3, you're factoring in that you are not R, big R, big R. So if you're big R, big R, none of your kids would then be affected.
Right.
Okay. Okay, I guess I know how we could say the problem. What is the probability that you wouldn't have to worry about a kid being affected? You wouldn't have to worry about any of your kids being affected if R was homozygous dominant. The probability that R is homozygous dominant is what? 1/3. So a third of the time you don't have to worry about anything. The other portion of the time you have to worry about your child being affected half of the time. Pat, you had a question?
Okay, that was part of the question. We just answered it. I was also wondering, like, let's say I'm a parent, so what is the percentage, like, of my child being on like, would that also?
What is the percentage of
Like 100, like.
out of your 100 kids that you'll have? You have to be specific in these problems to figure the calculation.
No, so my one child, what is the percentage?
What is the percentage of times that first child is going to be your first child is going to be affected? You just convert that fraction into a percentage. So it's going to be 33%, 33.33% of the time, roughly 33% of the time, your first child is going to be affected. Okay. You could also do permutations with this, right? What is the probability in this case? You can say, okay, given that say R and S plan to have 4 kids, what is the probability that 2 of them are going to be affected? You could actually now do the permutation. So these are sort of layered problems that you have to, you have to be able to read the pedigree. 2, get information from the pedigree. 3, calculate the probability of this and then do permutations for it. So as you can see, these problems can get more and more difficult. Okay, so here this is just showing you what I just showed you with the Punnett square. The probability is 2/3 because you no longer have 4 cells, you just have 3 cells of possibility because you know that individual is an albino. Okay, so key points, you can just go through and read the points, but I don't think on the PowerPoint that you printed out we had problems. I wanted to go through together and do a couple more problems. Okay, so here we go. Phenylketonuria is a metabolic disease that we'll talk a little bit more about in chapter 4. It's caused by a recessive allele, we'll call it little k. What is phenylketonuria? What do you not have the enzyme to do?
Can't handle phenylalanine.
Yeah, you can't convert phenylalanine into tyrosine. And you get high levels of phenylalanine that then spontaneously convert to phenyl something else, that you then get ketonuria, you get buildups, and you're concerned about that, especially with newborns, because, well, I'm getting ahead of myself. Anyway, don't worry about it. We're going to talk about that in chapter 4. Okay, so say 2 heterozygous carriers of phenylketonuria or marry and plan to have 5 children. Okay, here this is set up. Let's see if we can set this up. 1, or A, what is the chance that all of their children will be unaffected? We kind of already did this, right? 2, what is the chance that 4 of the children will be unaffected and 1 will be affected with phenylketonuria? It's just a little bit different one. We've kind of already done it, but we're going to go through, see how fast you can do it. So if you can just crank through this, you'll be in pretty good shape for these. C, what is the chance that at least 3 children will be unaffected? And D, what is the chance that the first child will be an unaffected girl? Okay, so it's a little more complicated, but not too much. All right, so let's take 3 minutes and see if you can do that. Okay, so is everybody good with this one? We're good. Just remember on the last one, you have to calculate probability of being unaffected, and the probability of being a girl. Okay, so now let's go to the last problem, and since we're getting pretty close to the end of the class, that's kind of crazy. We're going to try to get into 4 if we can, but I do want to do this last problem because it's a little bit more complicated pedigree one. And the pedigree ones are ones that people sometimes get confused about. Okay, so here's a little bit more complicated pedigree. I'll pull this down so you can look at it. Actually, yeah, so here you have 3 generations, right? What kind of mating is going on here? First cousin matings, right? H and I are first cousins. You want to figure out, let's just say, should we stick with albinism? Hemophilia is another one that we see. Let's say you want to, you want to calculate the probability of the first child of H and I, or the probability that they will have a child that has hemophilia. Okay? 3 generations, grandparents, fathers, or grandparents, parents, cousins, right? What is the probability?
This is a recessive trait.
What do you think? Carl says, ask me if it's a recessive trait. Now I'll flip it back to you and ask you, is it a recessive? Is it dominant? That's the first thing you have to do, right? You got to sort of assess these things. What do people say?
It's recessive.
Recessive, why?
Because if you look at the pedigree, you get the 1 out of 4 and the parents don't have it.
Yeah, so because E is affected, you know, both parents have to give E an allele, have to donate in their gametes an allele of a little a, or a little h, I guess in this case. Okay, why else? You have offspring where the parents don't show it. If it was dominant, right, one of the A or B would probably be affected. Remember, in dominant ones, the patterns, you see it every generation. Question?
Are we assuming that that it's not possible for the trait to be coming from outside the established family line?
Yeah, so that's a really good question. So some of the assumptions that we make about the pedigrees, right, is that when there's a marriage with somebody from the outside, if we're looking at a recessive trait, which we pretty much would be in that case, then we assume that that person is not a carrier unless there's information to the contrary. The question being, are we assuming that it's already there or that it's a new mutation, right? Unless there is evidence to the contrary, we're going to first assume that it's somewhere in the pedigree, right? If it were a new mutation, because you could make the case, right, that this is a dominant new mutation, a dominant new mutation, but usually those sort of things are rare enough that you're not going to see de novo 2 new mutations in one pedigree. So we're going to assume that we, because any pedigree you could just make the assumption, oh, it's a new mutation everywhere there's somebody's affected. That's not very satisfying from like a scientific perspective and not very realistic. And so we're not going to assume that. Okay, good questions. I don't know if we're going to assume that. Do we know it in one case? Maybe. Do we not know it in the other case? Maybe not.
But if there was a rare chance that they were homozygous dominant,
Yeah, because he's not a she and.
Okay, so since we only have a couple more minutes, let's go through and do it together. What's the first thing we always want to do on the pedigrees?
Write down what we know.
Write down the genotypes, right? Okay, so we know E is little a, right? How about F? Do we know F?
no.
What was one of the assumptions? Unless there's evidence to the contrary, we're going to assume that it's what? Big A. Good. Okay. Do we know? I can't write it all the way up there. Do we know big A, or do we know A and B? Do we know their genotypes? I can't write it all the way up there, but they're both heterozygous. So I would go through and write big A, little a, big A, little a. Okay. Any others that we know? Do we know C and D?
D has to be hetero.
D has to be hetero, right? How about C?
Hetero.
Hetero. All right, here is evidence to the contrary. Remember, we said if there's no evidence to the contrary, this one has to be big A, big A. But what is the evidence to the contrary?
That's the rule we're always.
That's a rule that's an assumption we're always going to have. Unless there's evidence to the contrary, it's big A, big A. But in this case, G is evidence to the contrary. G has to get a little a from both parents, right? So now what do we know? What do we know C is? Big A, little a, right? We know both of those. So we know everything in this second generation, or in this first generation, right? This parental, not the cousin, but the parental generation. We know all of those genotypes. Okay, in order for H and I to have an offspring that is affected, what do they both have to be?
Hetero.
They have to be carriers, right? Or hetero, right? So what is the probability that H is a carrier? Can we calculate that?
2/3.
Probability that H is a carrier? Oh, good. And how come it's 2/3? Because you know it's not.
It's not little a.
Yeah, you know it's not affected. So that's the probability, 2/3. Okay. Oops, I wrote it wrong. Wait, we stopped. 2/3. All right.
The way that we get that 2/3 again is that we just throw out
The little a,
possibility.
because we know that person is not affected. Okay? Otherwise, it'd be red up there. Okay, so now what's the probability that I is a carrier? So probability that I is a carrier? I is. So what's the probability when it's certain? 1. 100% or 1. Okay, 1. Okay. Given this individual is a carrier and this individual is a carrier, so we'll say big A, little a, big A, little a. These are both those H and I, right? What is the probability that the child will be affected with albinism? 1/4, right? So here, probability, given all of those, probability of, we'll just say CA that the child is affected is 1/4. So how many events have to occur? We'll just say 3 events. This and this and this have to occur for that child to be affected. How do we do it? How do we calculate and? We multiply 2/3 times 1 times 1/4 equals 2/12. Is that right? 2/12, which equals what? Reduces down to 1/6.
What's that bottom one? The probability of what?
Probability of, it's kind of a long sentence, probability that the child will be affected, child affected, if both of these two individuals are carriers. So that's basically this right here, what we just did. It's just a way of shorthanding. All right, how many of you got that answer right? How many of you got it right before we did it? All right, I think you should all look into being genetic counselors because you're well on your way to the big bus. All right, question.
Yes.
Oh heck, maybe come up. I can't hear you. I can't hear you. Oh. This raucous first row.
Whatever, I already did. I'm gonna be a patent troll. I already did. Oh man.
I got the same answer, but I did it differently. So
Well, open up like a dental family practice.
Yeah, so a carrier.
And you take that go.
So I did that.
Both entomology museum on the side.
2/3 yes, this is a carrier.
There you go.
Half of that.
Yeah. So,
So I did 1/3 times 1/2.
oh, so you just factored in. You factored in. Let's see. So you got this one. You said is a carrier, and half of its gametes are going to be passed down. So that's factored into what's happening here, right? And this one to be, you said.
2/3 chance it's a carrier.
Uh huh.
Half of its gametes.
No. So you just had this 1/2 here. You said 2/3 chance of carrier, right? 2/3, and then half of those will be passed down. So you did it. Same thing. You just basically separated out 1/2 times 1/2 rather than just that 1/4. You see? So yeah. So go look at that. So, yeah, you just skipped over this part really by saying you just looked at it a different way. You said, okay, half of this individual's gametes are going to be little a, half of this individual's gametes. That's the same thing they're saying over here. For that to happen and that to happen, you just times those two. Or this is just a graphical way of saying that, getting the 1/4. Okay.
Okay, so I understand this part. I understand why this is 2/3, but I'm kind of confused. So we're assuming that outside is gonna be AA, but it's not gonna have any.
It's not a carrier.
Okay, so take it,
Because they're,
So then all of the offspring, what are all the genotypes of the offspring going to be?
big A, small A. Okay, so when we do this,
So after that 1 is right here, and you know that genotype.
it's 100%.
Yeah, 100% chance that that genotype is going to be big A, little a.
And then, where did, so the 1/4.
So 1/4, okay, so let's just say, all right, let's say we figured out the probability that this individual is going to be a carrier. So let's just pretend, okay, we're now assuming, we're assuming that it's big A, little a, okay? So assuming that that's big A, little a, what's the probability that these two are going to have an offspring that is affected? Yeah. Okay, so 1/4 here. We assume that, but we know that it's not necessarily a carrier. What's the probability that it's going to be a carrier? No, that it's going to be a carrier. That's at 2/3. Right, between that and that, that's a 2/3. All right, make a little more sense? It's not going to make total sense until you start just